Okay, we should go ahead and get started. Good morning, good afternoon, good day, everybody. Welcome to the first day of May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. Oops, and I hear myself on YouTube at the same time. So welcome everyone. We are very excited to see so many of you joining this program. We have fantastic instructors who worked very hard preparing the materials for you. And even though we cannot meet in person, I really hope that this will be a good experience for you. So normally we would be in Boston on Northeastern campus. So I hope that we will regardless be able to interact through the online platform. So before we start, let me do a little bit of housekeeping items. So we're using the webinar platform primarily for security reasons, so to avoid unpleasant surprises. However, we do hope that this will be an interactive experience. We do hope that you will be all active participants. So you see the chat button in the webinar. So the chat is used primarily for logistics reasons. I can't hear you, you know, where is the link? It is too slow, too fast, or something like that. But for science-related questions, please use the Q&A button, because this will allow us to streamline and triage the questions and direct them to the presenters at the right moment. We can also give you voice and screen sharing privileges if we have an interactive discussion. And finally, we have teaching assistants who are ready here in case someone needs more hands-on or individualized help. So for example, if you are stuck with some downloads or have some questions which really set you back and do not allow you to move forward, teaching assistants will broadcast separate Zoom links for you, where you can go and join the TAs in the separate offline link and work things out. And so please speak up if this is the case. We will make sure to accommodate everyone as much as possible. So this is the plan. So if you have logistics questions, again, please put them in chat. Otherwise, we will have them in the Q&A. And please do not be shy to speak up. It is really meant to be here for you, so that you can learn as much as possible and you gain as much as possible the information that is actually helping you in your own projects. So without further ado, let me start by welcoming our first opening keynote speaker, Brian Shorroll. It is my great pleasure to introduce Brian, who is a great scientist and a great educator as well. So Brian has a PhD from University of Washington under direction of Mike McCoss. And then Brian moved to the Institute for Systems Biology in Seattle as a translational research fellow. And I understand that today is his day one of being assistant professor at Ohio State University. Congratulations, Brian. This is really exciting. We expect great things from you. And I should also add that probably many of you have heard of the company Proteum Software and the software tool called Scaffold. So this is, of course, due to Brian and his colleagues at Proteum Software. And so I hope that all of these great things that have been done in the past will now, Brian will continue making great contributions. So I'm really excited that Brian joined us today, and I look forward to hearing from him about the introduction to DIA. Thank you very much. Brian, we cannot hear you very well. Can't hear you.
What happened?
Can you say something?
Okay.
It is better. It's just very faint. I think we need to tune the volume up. Maybe you can just hold the microphone closer.
All right. Is that better?
Yes, it is better.
Okay, great. Sorry for the, I don't know what happened in between there. Okay, so as Olga says, I'm going to give you a little bit of an introduction to data independent acquisition. And then after this, Lindsay is going to give up sort of a follow-up, a practical tutorial on how to start to interpret this type of data. So I'm excited for that. You should definitely see both of these sessions side by side. So as Olga said, I'm just starting today my appointment at Ohio State University. So I have an email address down here, but I also have my personal email just in case if you need it. Also, the slides for the this session are actually on the website, but these are Creative Commons licensed. So if you want access to the original PowerPoints, feel free to email me then, and I'll try to get them to you in a reasonable way so that you can look at how the slides were working and then dig into them a little bit more deeply, or potentially present that material on your own afterwards. So just to kind of start off, I like to give a slide about speaker disclosures just to explain to you where I come from. So as Olga said, I'm now an assistant professor at Ohio State University, but I also have a founder's position at Proteome Software, and I'm a shareholder there. Most of my research funding comes from the NIH and from a little fund associated with IARPA. So with that in mind, I kind of wanted to start us off easy. So just to explain to you a little problem that I had been working on recently, I've been interested in studying how infection works in cells. And so I've been analyzing proteins from viruses in human cells. And so how would I do a problem like this, right? The first thing that I would do is I would take those proteins and I would linearize them. I would stretch them out and denature them. And then I would digest them with a digestion enzyme, in this particular case called trypsin, and cut them up into little pieces that make it easier to analyze with a mass spectrometer. And then finally, I would try to analyze that list of peptides that are generated. You know, most of those peptides are going to come from, in this particular case, the human cells, and the peptides that I'm interested in are essentially like needles in a haystack. They're hard to find. So in a problem like this, which is in a sense, very similar to virtually every type of proteomic problem, rarely are we interested in the most abundant albumins of the world. We're really interested in the stuff that's difficult to see generally. So in these particular cases, we have a bunch of tools to be able to do that. So I want to lead you through some of the ways in which we analyze this type of data. And the most common way in which we analyze this type of data is called data-dependent acquisition. So here in this framework, we're analyzing peptides as they elute off of an HPLC, separated in time over retention time, and separated in mass by the mass spectrometer in precursor space. So this would be a circumstance where each one of these peaks represents a different type of peptide that is in my sample at any given time point. So the way DDA works is that over time, we see a peak come up, we select it for fragmentation, we collect a fragment spectrum, and then we stick it on an exclusion list where we ignore it for a while. This helps us be able to dig deeper and deeper into the proteome. And so then we'll go to the next peak, and then the next peak, and then the next peak, and the next peak, and the next peak, and we'll sequence all of these guys and give us an idea as to what's going on at each of those different peptides. So how does this work in the mass spectrometer? We collect these precursor scans like we showed you before and pick peaks out of this. In this particular case, I'm showing you the top 12 peaks that the instrument would pick based off of intensity. And then all of the rest kind of get left out. And then again, all of these 12 will get stuck in an exclusion list so we can sample them over time, sample other peptides over the same time period. But there are some complications with this approach. So right here, I'm just showing you kind of a screen capture of what 60 seconds of peptides looks like. So this is, if you're looking at a precursor scan over time, you can see the peptides rise and fall over time. And so you can watch them with their relative intensities go up and down. And there are some regions of this proteome where you see these peaks that rise up and down, that they look like normal peptides, but they're just never going to get to the threshold at which we would ever try to send them for a sequencing scan. So there are lots of these types of peptides that are really valuable things that we'd be interested in detecting that we just won't ever be able to see. So if I take this, again, this 60 seconds of peptide screenshot and sort of show you all of the peptides that, in this particular case, this is on a Q Exactive. These are all peptides that are above, that I've colored here, are peptides that are above 1E7 in intensity. So that's a very high threshold in terms of its peak area. That's much higher than we usually set for our normal dynamic exclusion thresholds, which tend to be around E3 in terms of so 4 orders of magnitude separation between them. So there are a lot of peptides. These are all of the peptides that are in that range, but already there are more peptides in this time point of just 60 seconds than we have scans, than we have MS2s that we can allocate towards measuring these types of peptides, already within E7 in this particular case. So we're saturating in terms of collecting MS2s very quickly. Most of the time we saturate almost instantaneously. And so let me show you what that means. So up at the top here, I've got a normal sort of tick trace where I've got retention time over time and then precursor tick on the y-axis. This is just showing you where the density of peptides are eluting off of my mass spectrometer. And you see quite quickly on the bottom graph where I'm looking at the number of MSMS collected in purple in a normal DDA scan, we hit the maximum point, the maximum number of MSMS measurements almost immediately, very early into the gradient. And after a certain point, we still are collecting huge numbers of MSMS even out to the very end of the gradient. Now, if we look at the number of peptides detected, so the number of PSMs measured in this time period, what does TIC stand for, Jennifer? Great question. TIC stands for the total ion current, which is basically a measurement of the amount of ion signal that we're seeing in the mass spectrometer at that time point. So if that signal goes up, then we have more and more peptides at that time point. It's instead of measuring a single peptide, we're actually summing all of the signal from all the peptides. Does that kind of make sense? And PSM, what does PSM stand for? Peptide spectrum match. So these are the number of detected peptides that we're actually seeing at any given time point. So in this particular case, at time point 30, we're detecting about we're collecting a full number of MS spectra, but we are only detecting about half of those spectra as actual peptides or assigning to actual peptides. So one of the things that you can see that's really interesting here is that as you get into a sparser part of the gradient, that's where you're seeing fewer and fewer actual peptide signals from the tick trace. We actually identify a higher percentage of peptides in our total number of peptides as those PSMs. So the green bars are higher than in that early section where you've got lots and lots of peptides based off of the amount of tick signal there. And so what's going on here? Actually, what we're seeing here is a circumstance where the measurements we're making aren't actually of individual peptides. And we are actually making collecting MSMS spectra that contain multiple peptides at the same time. And because of that, it makes it very difficult to make detections from those peptides. And this is just because when you have more and more peptides, the sort of space that you're able to sample gets more and more congested. And so when you select out a particular mass for MSMS, oftentimes there can be other peptides at those same points. So how do we sort out impurities by seeing these graphs? This is an excellent point. So all I'm showing here are +2 and +3 signal peptides. So there are impurities that are +1s that are usually sugars or other types of small molecules that could definitely be a potential problem here, but that's not actually what's going on here. The instrument automatically filters out any peptide that doesn't have a +2 charge or higher. So we know that these are measurements that we should be able to detect peptides from, but they're just too complicated to be able to make those measurements because they're coming from multiple peptides at the same time. So DDA, I think, is a really excellent tool for doing this type of measurement. And yes, you could increase retention time for doing this type of measurement. That's a good question, Christopher. But that said, the more you increase retention time, the more you have to spend time on that specific sample and not on other samples at the same time. So you can stretch it out further and further, but that still also doesn't make it such that if two peptides have somewhat similar chemical properties, they will still fall on top of each other, even if there is space between those peptides and other peptides. So you can make the problem a little simpler, but it still has a asymptotic potential. So just sort of thinking about DDA, we are making these fragment ion measurements, and we're collecting MS2s about them. And yes, we can do some little tricks about decreasing isolation window and all that kind of stuff. But these still, realistically, still let in other types of peptides at the same time. So unless you're using an orthogonal type of separation, say, for example, ion mobility, you're almost always going to get these very convoluted spectra where you see multiple fragment ions from other types of peptides at the same time. One way to think of this is anytime you see noise in your MS2, that noise actually comes from other peptides in the background. So there's definitely a lot of interesting signal that exists that we're not able to capitalize on because of the way we're selecting precursors and trying to assign a single peptide to each spectrum that we're measuring. So another strategy for trying to do this is trying to analyze samples with a technique called PRM. And if we're interested in monitoring these low abundant peptides, so for example, in this particular case, you can see that the dark blue and the light pink peptides were just never sequenced for MS2s. We can actually target specific masses that go after individual peptides that we're interested in. And rather trying to collect a single MS2 to represent that peptide, we can actually collect MS2s over time, one after another, sequentially over time. And this kind of gives us a way of measuring the peptide at its apex, and by being able to see a peak shape associated with it, we can be very confident that that peptide is actually present. We also guarantee that we're going to select the peptide of interest. So for example, in this particular case, that blue peptide. So this is a way for me to select out, for example, all of my virus infection peptides to be able to measure those very well. But one of the things that is a potential problem is I'm interested in measuring all of the viral peptides, all of the sources of viral peptides, not just selected ones that I'm going after. And so, yeah, Julia, you make a very good point. We do have a limit on the number of masses that we can assign in a PRM experiment. Typically, in a normal PRM experiment, we're only really able to analyze something between 150 and maybe 300 different peptides at a given time point, corresponding to maybe 50 or 100 different proteins. So we're not able to measure an entire proteome here. We're only able to measure a handful of peptides belonging to a handful of proteins. So that's definitely a complication of PRM that makes it limited in some circumstances. So just to kind of reiterate, PRMs are really great for making deep measurements, but they are a little bit complicated in terms of trying to measure an entire genome. So if I'm interested in measuring an entire genome, we actually need to ask the question, is it possible? Are there mechanisms to target every single protein? And there are some kind of clever ways that you can run PRMs to be able to do that. But what I want to do is talk to you about another way of thinking about this, which is data independent acquisition. So just to kind of get to a content slide, here I'm looking at DIA, and I'm going to talk to you about how we're acquiring this type of data and then what types of measurements we're trying to make, and specifically thinking about it from a PRM perspective. Because instead of trying to do a DDA-like acquisition, we're actually trying, with DIA, we're trying to do PRM-like acquisition, just in a global way. Then we're going to talk about how we're going to analyze DIA data with libraries, some of the complications around measuring DIA data in experiments, in certain types of experiments. And then we're going to talk a little bit about some of the best practices for setting up a DIA experiment in your lab. And then finally about how we're going to analyze data if we only can acquire DIA data, which is oftentimes the case, and how I frequently am acquiring data these days. Okay. So just starting off at the beginning, what is DIA? And it's basically trying to take a PRM and expand it out to a global acquisition strategy. So again, thinking about that PRM type of measurement we've got here, if we're trying to section that off as if we're looking at it from a DIA perspective, instead of selecting a specific peptide to measure, we're selecting a precursor mass window. So for example, here we would be selecting the mass window from 475 to 500 MZ and from 500 to 525 MZ. Okay. And so what's going on here is that we are actively co-fragmenting multiple peptides at the same time that come within the same wide window in this particular case. So the MS2s that we're collecting, the individual mass measurements, are extremely complicated. They come from oftentimes 20 to 100 to even a couple hundred different peptides at the same time in the same scan, in the same MS2. But one advantage here is that we get time dimension information. So we can monitor these fragment ions over time as they rise and fall. And so then the goal of a DIA experiment becomes to select out from those complicated interfering signals, which are the fragment ions that are associated with a specific peptide. And then all of a sudden we're looking at data that's much more similar to PRMs, and we can think of it from a targeted perspective, where we're making these measurements of normal fragment ions across time. In these particular cases, when we look at these things, we're thinking about peptides eluting over around 30 seconds or 60 seconds or therefore. So what are the kind of constraints that we're thinking about here? So there are some compromises that we're trying to deal with, and one of them is the fact that each of these peptides elutes over 25 seconds or so. And so the instrument can only scan so fast without sacrificing those types of measurements. First, we need to be able to collect good MS2s. And so good MS2s, it takes about 10 to 20 hertz on Orbitraps. You can get up to around 100 hertz on some TOFs, but as you start to speed up, you actually are dealing with fewer and fewer ions at any given point. So the actual MS2s that you're collecting are not quite as robust or not quite as accurate. So you're actually limited in terms of the scan speed at which you can acquire data. And usually we're thinking around 10 to 20 hertz, at best around 100 hertz. And the other aspect of this is you really need 6 to 8 measurements across a peak to be able to describe the shape of that peak. So you need to collect a lot of scans to represent every single peptide. And so there are a lot of compromises that we make relative to PRM. And one of them is that we have to accept much higher levels of interference by widening our precursor windows, as I said. So rather than targeting a specific mass, we're actually targeting a range. So in this case, 25 M/Z window. It lets in a lot of different peptides simultaneously. So we have a lot more interference in our measurements. The other aspect is that we have less time to sort of spend on each mass spectrum. So we can't just like sit and acquire on that mass spectrum for half a second or something like that to be able to get the best signal. We actually have to run through these scans relatively quickly. So as I said, in the order of 10 hertz to 20 hertz to 100 hertz, in that kind of range. So the other aspect is that we kind of need to limit what we can measure. So rather than being able to measure any type of peptide, usually we have to think about a narrower window scheme where we're only measuring peptides from 400 to 1200 or 400 to 1000 or something like that. So what do these compromises actually look like? So what I'm showing you is measurements that I collected on a QE for PRM, where I get really good signal on the across the peak for these 4 transitions, 5 transitions. You get really nice signal. And if I look at that same data with DIA, you know, I get I run up into some different kind of constraints. You know, one of them being that there's more interference and that there's a lot lower signal to noise, and also there's less points across the peak. But realistically, these types of measurements are almost identical. You can see that the maximum intensity of the PRM, again, goes up to 10 times 10 to the third. And again, you're seeing the same type of signal in the DIA experiment. It's really the same type of measurement. So in that regard, we can actually analyze this type of data, thinking about it as a targeted experiment, and we get a lot of benefit out of being able to reuse some of those concepts. So Lindsay later is going to show you how to analyze DIA data using Skyline, as well as some other potential tools that center around this sort of targeted perspective of trying to interpret this data. So what does DIA data acquisition actually look like? So as I said, it's comprehensive acquisition where we're acquiring a precursor spectrum, and in this case from, say, 400 to 1200, as was with the original SWATH papers or DIA papers. And you collect a fragment ion scan versus a reasonably short range in that precursor scan, and then another, and then another, and then another, and eventually you scan through the entire window. You'll go down and collect another precursor scan and then another set of fragment ion scans. And the goal here is that you want to get around 10 points across the peak for a 25-second peak. So you need your window width. So that's the distance between each fragment ion scan and each other fragment ion scan to be around 2.5 seconds. So, so they need to happen in relatively fast order. So all of those, the precursor scan and all those fragment ion scans have to happen in that same type of cycle time, around 3 seconds or 2.5 seconds. So let me take that data, that graph, and kind of compress it back. So now I'm just sort of shrinking it back so that you can see it a little bit easier. And if we zoom into one of these particular fragment ion scans from 400 to 75 to 500, we can look at this peptide here, LSGG, that we can see a really nice spectrum for. This would be as if we were thinking about it from a DDA perspective. And the reason, Hugo, why we're looking at 2.5 seconds is, again, we need to get 10 points across a peak that is 25 seconds wide. And so if you divide 25 seconds by 10, you get 2.5 seconds. Okay. So if you think about this, we're actually acquiring scans, multiple scans, one after another, to describe the shape of the peak. So rather than just looking at a single MS2 showing this peptide, we actually can see lots of MS2s describing this peptide. So we have tons of information in this sort of 3-dimensional space where we're thinking about mass over charge and retention time, not just mass over charge and intensity. So mass over charge, retention time, and intensity. So again, if you look at this from the angle, you can actually extract out all of the fragment ions that are associated with the peptide that we're interested in and make a measurement like we would normally do with PRMs. And so this approach allows us to kind of ignore all of the peaks that are the bigger peaks, the gray peaks that are essentially interferences from other types of peptides. Okay, so there are some complications here, and we've already alluded to a bunch of those. But thinking about this again, within the framework of DIA, all of those mass measurements over and over, one important thing is that when we're making these measurements, the ions that we're measuring are actually not centered in that window necessarily. They can be anywhere in that window. So they can come from the very early part of that window, so closer to 475 or closer to 500, and they can be of any different charge. So when we're collecting an MS2, the ions that we're taking a measurement of aren't centered, and they're also not collision energy optimized for that specific mass over charge. So the fragmentation that we get is not exactly the same as if we were doing DDA, where we can sort of optimize what the appropriate collision energy is on the fly for any specific peptide, because we're isolating a specific peptide or trying to isolate a specific peptide in a DDA experiment. So that's issue number one. Issue number two, as we've already talked about, there are usually multiple peptides that we're trying to measure at the same time, co-eluting at the same time. So the fragment ions can overlap and sometimes can be exactly the same fragment ions, particularly in cases of low mass fragment ions. The third thing is that we have suppression at the edges of windows. So for example, at the beginning of that 475 and at the end of that 500 mass range, we actually have essentially a trapezoidal transmission efficiency. So not all peptides are actually put into the mass spectrometer in the same type of way. Or at the same intensity. So for example, that red peptide on the end might not get as much signal actually put into the mass spectrometer as that green peptide in the middle. Okay, so so let's look at a DIA data set. So what does this type of DIA data actually look like?
To interrupt, there is a question. Yep. Could you comment on the choice of the specific value for the ppm?
Oh, so in that particular case, when we were looking at a particular peptide, that was just the ppm of the mass accuracy for that peptide measurement. So that's really all it represents. It doesn't actually represent anything. It's just what was the ppm accuracy for that mass measurement.
Sorry, there was one more question, Brian, that I thought was really good. I think you're going to get to it later, but what do you think about isolation windows overlapping, having more peptide identification? So overlapping DIA windows, more identifications? Yes, no?
Yeah, so we're definitely going to talk about overlapping windows later on. So that's a fantastic question. We'll get there, definitely. And the last question, can I collect DIA data for discovery using the same data as for a targeted experiment? So what we are trying to do is actually do discovery style experiments just with essentially targeted data, right? So we're trying to treat this as a merger between those two different approaches, between a normal discovery acquisition, but specifically from a targeted framework. So the type of data that we're acquiring looks like targeted data rather than looks like DDA data. Okay, but it's still trying to be a discovery platform like DDA. Okay, so what does some of this DIA data look like, and how messy actually is it? So here I'm looking at a specific HHAA peptide from superoxide dismutase in this particular sample. And so if I show you all of the extracted fragment ions across time, each one of these peaks, each one of these colors represents a different fragment ion in that particular peptide. And I'm just extracting all those fragment ions out over time. So for example, if that first fragment ion or those first couple of fragment ions have a mass of say 475, here I'm extracting out 475 across the entire retention time to show you what's going on. So the first question is, where is this peptide? It's actually pretty hard to figure out normally, sort of naively, where this peptide is. And so let me just show you, it's right here. It's not the most abundant fragment ion associated with this peptide. It's actually one of the smaller fragment ions. But the way we know that this is the right peptide is we can look at the combination of multiple fragment ions together, and the presence of multiple fragment ions, even if you sum up these signals, they're going to be less than the most abundant signals. But they are, there are a lot of signals all coming out at the same time. So we can say, because all these fragment ions trace together, we can be pretty confident that this peptide is here. Now, this looks like a great example. Obviously, DIA works in this particular case. But let me show you this other peptide, this FNGG peptide. This is from, again, superoxide dismutase, exactly the same protein in exactly the same sample. So these should be equimolar in their abundance. So they're on the same column at exactly the same amount. And again, where is that peptide? It's actually in this grass here. But when I show you the region around here, it's kind of complicated. You know, actually, what is the peptide that I'm actually looking at here? Is it the peptide on the left, or is it the peptide on the right, or is it the peptide in the middle? It's really difficult to say what is the right signal associated with a specific peptide. And so let me just show you, it's this particular signal, even though that's the one that looks the messiest. But how do I know that? And I know that because I can use information from previously acquired data. So I can use information in a library where I know the fragmentation of this particular peptide from a DDA experiment, a past DDA experiment, and I can look in my sample at that same time point and extract out all the fragment ions associated with that peptide. And lo and behold, it looks beautiful in terms of the integrations for all of those guys, where I've got high Y8s and Y9s, Y2s. I've got, again, a nice ladder from Y10, 11, and 12, and then all of the rest of the ions are lower than that. So I can be really confident that this is the right peptide based on those fragment ion intensities. But when I actually look at the underlying data, this is what the underlying data actually looks like. This is at the apex spectrum. This is a signal at the apex spectrum. This is the highest signal for this peptide. It's still hidden in the grass in this complicated spectra where I'm actually measuring, again, 20 to 100 different peptides at exactly the same time. So if you were trying to interpret this from a DDA perspective, you would end up kind of like making it would be very difficult to make this interpretation of this as being the specific peptide being present here. It's actually a much lower signal. Okay, so, and we'll talk about, Raja, we'll talk about lots of different strategies for trying to think about analyzing this type of data. We'll get to that in a bit. But so one of the things that I can do here is looking at the fragment ions that are associated with this particular peptide. I can say that some of them are interfered with, say the ones in red. Those are just kind of not very useful in terms of making this particular measurement. The ones in yellow rise and fall with the peptide, but they also show some level of interference as well. The ones in green, we can say, you know, those particular fragment ions, even though they're not the most intense fragment ions, because they rise and fall with the shape of the peptide, we can be really confident that that's what's actually going on here, that we actually are seeing this type of peptide. So Bowen, we're going to talk quite a bit about a library generation in a bit. So realistically, this DIA data is actually extremely hard to interpret when you're thinking about it from this way, this sort of naive way of trying to extract out the data. So we have a bunch of new approaches that we're going to get to for why we would actually, how we're going to extract this type of data out. But the first thing that I wanted to ask is, why even bother collecting this data if it's so hard to interpret? And there are some really interesting advantages. So when we're actually trying to analyze DIA data, we see lots of interesting features in the data that we don't see with DDA, because we end up making mistakes oftentimes with DDA. So one of those, for example, is getting an early DDA mass trigger. So for example, this particular precursor mass, we see, again, rise and fall as we would normally expect to see. We would trigger it from MS2, but there's actually no signal for this particular peptide. And we can see that there is another peptide that comes out just a hair later, less than half a second later. We can see that, or half a minute later, we can see that second peptide again come out with great MS2s associated with it. But we would never measure this with DDA because we would have stuck that peptide on a dynamic exclusion list. So another aspect of this is that with DDA, we find that the precursor signals oftentimes can be very complicated. They can have a lot of interference because it's only 3 measurements that we're able to make. We're able to make a monoisotopic measurement, a +1 and a +2 measurement. Again, the blue is the monoisotopic, the purple is the +1, the red is the +2. These are typical skyline colors that you'll be comfortable with later after Lindsay's presentation. But here we can see that there's these precursor signals. While they kind of line up with the peptide, they don't line up with each other. That indicates to me that all of these signals are heavily interfered with, and they actually come from multiple peptides at the same time. So if you try to integrate the precursor signal for this peptide, you'd end up with kind of a mess in terms of the actual measurement that you're making. And the third one is even the most interesting of all. We see a lot of peptides, actually, a reasonably significant number of peptides that don't have any precursor signal at all. And so these are peptides that just go completely blind in the mass spectrometer, but we can see really great fragment ions associated with those particular peptides. And so what's kind of going on here? One way to think of this is if you're acquiring normal DDA type of experiments, you're taking all of the peptides that are coming off the column at exactly the same time, you're passing them through a quadrupole. And then in this particular case, I'm showing you with an Orbitrap, but the same kind of principle occurs in other types of instruments too. In an Orbitrap, you take those peptides and you put them into an HCD cell where you do some fragmentation. And you make the Orbitrap measurements of those particular peptides. You'll see a bunch of those. So in this particular case, we're actually just using the HCD cell to store the peptides. So we can accumulate in this particular case up to about 1e6 in a QE number of peptides, so 1 million peptides that we can actually analyze at a given time in a given scan. So if you've got a nice representation of your 3 peptides amongst those million ions, then you can get really good measurements for their precursor scans. But realistically, if there are fewer and fewer of one particular population, you might not have any of those ions actually in that 1 in a million HCD cell for being able to collect ions. So if we're trying to collect 1 million ions, and we're actually measuring something that is 3 or 4 orders of magnitude, 5 or 6 orders of magnitude down, there might just not even be any measurable ions associated with the peptide that you're interested in, and all of a sudden your signal for that particular peptide disappears. But this is actually one of the reasons why techniques like Boxcar work, is that with Boxcar, you're actually using the quadrupole up front to filter out ions that are not associated with the region that you want to look at the precursor space for. So if you only measure, in this particular case, the precursor mass around the orange ion, then you're going to definitely see measurements for that orange ion, which is why Boxcar is so effective at being able to see good precursor signals for peptides. DIA is essentially the same thing, just a much earlier approach. So the advantage of this is that, as opposed to just getting an accurate precursor mass with Boxcar, you actually get fragment ions associated with those peptides. So we're just doing fragmentation in the HCD cell and measuring all the fragment ions for those selected ions. So in a sense, Boxcar and DIA are actually really similar concepts. It's just whether you're collecting precursors or fragment ions associated with those peptides. Okay, so this is why you would want to do DIA versus a DDA. There are many different sort of reasons for this, but why would you want to do it over PRMs or SRMs? And a lot of that actually has to do with the sort of workflow in which you're analyzing PRMs. In this particular case. So Gabriella, actually Boxcar you wouldn't want to use to build a library, because you actually need a library to analyze Boxcar data. Because you aren't necessarily collecting fragment ions, you only have precursor ions. So you don't necessarily know what the particular peptide sequence is, you just know what the precursor signal is, because that's all you have time to do. Now there are combinations of Boxcar plus DDA or Boxcar plus DIA that kind of get that make Boxcar be able to analyze data in the same, or interpret the data that is collected for quantitative work in the same experiment. But those are more complicated types of data acquisition. But if we're thinking about PRMs, one of the things that we have to do with PRMs is we have to think and sit down and think about what peptides are we going to select, because we only got 100 peptides or 300 peptides that we can actually measure at any given time point. So we have to think about which peptides we're going to measure. We have to select those particular peptides. We have to figure out when they're going to elute, and we're going to have to come up with which transitions represent those peptides best. And only then can we start acquiring data for those peptides. And so oftentimes what we do is we acquire a preliminary experiment, and then we say, actually, we needed to measure these other peptides at the same time, because they belong to a different pathway that is also really important. And then we got to go back and select some new peptides and do some more scheduling and some more refinement. It's actually a long process to be able to build a robust PRM or SRM assay. It actually takes months to be able to make these things very robust and very accurate. And so good target experiments take a ton of time because of this sort of got to think a lot before you actually acquire data. With DIA, we actually have this interesting alternate problem, which is that we can actually acquire data up front. We don't even need to think about which peptides we are interested in monitoring, because we're collecting them across all of the peptides from 400 to 1200 or 400 to 1000 MZ. And then we can come up with our hypotheses. We can do our peptide selection and chromatogram extraction. Instead of doing these refinements, we can actually just think of it as extraction out of the DIA data and this transition refinement out of the DIA data after it's been acquired to decide which transitions are actually useful for making those measurements of those particular peptides. And then we can go back and we can circle around on the data analysis rather than trying to circle around and have to reacquire new data after one after another. Okay, so now that we've started to think about what DIA data is and how sort of we can roughly think about it from one experiment to another experiment, let's look at how do we actually do some analysis of this type of data. And we typically are analyzing this data with libraries, but let's reason why we actually need to do this. So with DIA, this issue of peptide selection is really important. So you could think about going from the most broad, you could actually look at all possible tryptic peptides associated with all possible proteins in your proteome. That would be sort of the most broad DDA style approach, all the peptides in your FASTA database. Down to, you can do the predicting for all the hyper-responding peptides. You can look at stuff that you've already seen in DDA libraries. You can build DIA-specific libraries, which we'll talk about at the very end. Or you could actually just look at peptides that are identified by DDA in your specific samples, which is maybe the most limiting approach. So why can't we analyze all tryptic peptides in the proteome? And let me give you kind of an example. Here is a protein that is a transcription factor called Cas-Z1. And these are the transition responses. And again, this is on the log scale. So this is the actual responses in signal from the first peptide, the most abundant or most highly signaled peptide, and the lowest peptide. And this has to do with response in the mass spectrometer. Certain types of peptides ionize well, certain peptides release from the protein with trypsin well, and other peptides don't necessarily release as well. And so what we're looking at in this particular sample, or this particular protein, Cas-Z1, is 3 orders of magnitude, 1 to 1,000, or in this case, even more than that. So it's 2.5 orders of magnitude in this case. Response between the best responder and the worst responder. And this is actually really common due to ionization. If we look across, in this particular case, I was looking at almost 1,000 different proteins in terms of their best response and worst response in equimolar proteins that were, in this particular case, synthesized for this very problem. So we knew we had the full-length protein and we had equimolar peptides for every single one of those peptides on column, or at least available to trypsin at that point. And we see 3 orders of magnitude is actually the average of 3 to 4 orders of magnitude is the average separation between the top responder and the worst responder. So realistically, in a DIA experiment, we're going to see the first couple of peptides, the most easy to see peptides, and all the peptides at the bottom are just not even worth thinking about. They're not even worth considering because they are such poor responders. So that's why you actually, when you're looking at a peptide sequence, you actually are only seeing a handful of peptides across that protein, not all the peptides across that protein. Okay, so that kind of gives you some intuition for why you can't look at all tryptic peptides. But so most of the way we think about this is from thinking about libraries, specifically around DDA spectral libraries, because that's sort of the most common way to think about this type of peptide. So one of the ways in which we can do this is, so again, just thinking about what we talked about earlier about extracting out fragment ions, where we're looking at the library spectrum versus actual spectra. The way in which we try to make this type of detection is we monitor over time. We do this sort of extraction of all of the fragment ions out over time. So this is, again, all those different peaks. And each time point, we actually score the library spectrum versus the MSMS spectrum that we've acquired, or the fragment ions that we expect to see for that specific peptide. And so we expect that score to, be around a baseline, and then at a certain point spike up. And we hope that at that certain point where it's spiked up, that's actually an indicator of the particular peptide. So if you are interested, for example, Simi, in analyzing tryptic peptides and semi-tryptic peptides, so very complicated types of peptides, or for example, phosphopeptides or unusual types of modifications, those circumstances, you would need to basically build a library specific for those types of peptides and look for those specific peptides as you're going. Okay. So how do we analyze this type of data? Specifically, a lot of you guys have seen sort of target decoy analysis for computing false discovery rate, where you actually have, you search for a forward FASTA database as your normal FASTA database, and you've got a reverse FASTA database that you search at the same time. And the advantage of searching this as a combination is that you can say, well, you're not really sure about any particular match to the forward database, but you can know that any match to the reverse database is definitely wrong because those peptides don't exist in the real world. So that kind of gives you a way to say, what do bad peptides look like? What are the scores for bad peptides? And how far away, how much better are the scores for the forward peptides than than these reverse peptides, as a way to judge how confident you are in that type of measurement. You could do the same type of thing with library spectra, actually, where you've got sort of target spectra, and then you can construct decoy spectra. And the way we construct these decoy spectra is we take the sequence and then we reverse the sequence except for the termini. So we keep in this particular case, the V and of the vape peptide and the K the same, and then we reverse all of the sequence in the middle, and then we keep the same ion intensities, but we move the masses around and potentially the mass errors around for those particular fragments. So you can see that the intensities are the same for the ones that I've selected out, but their masses are a little bit different. That helps us be able to say this is a decoy rather than a normal spectrum. Okay. So this is one approach to analyzing data. This is called the peptide-centric approach, where we're extracting out all of the fragment ions that may be associated with that over time. And there are some great advantages for it, but I want to give you another idea for how you could also interpret this type of data. And that's using a method called DI-Umpire, and there's a bunch of other types of methods that try to do the same type of thing. But the idea is basically they try to make pseudospectra from DIA. That's where you've got DIA data over time, and you want to extract out what a specific fragment ion, what a specific fragments are associated with an individual precursor are. And so they look at the sort of extraction for a particular precursor that they say that is of interest, and then they look for any fragment ions that have that same retention time characteristic, the same rise and fall as that precursor peak. Now this runs into a lot of the same problems because it requires that you actually have a precursor signal that is accurate and representative of the shape of the peak, which we talked about earlier as not being necessarily a perfect scenario and some of the things that DIA can resolve. But it is an approach where we say these particular precursors can be assigned to these specific fragment ions, and that lets us compute a pseudo spectra of fragment ions that have the same rise and fall as that individual precursor. And then we can send those guys through a normal DDA-like platform where we're analyzing data with Mascot or with Proteome Discover or something like that. Okay, so what are the advantages for this sort of spectrum-centric approach is that we can use it like normal DDA-based methods. And so here we can take our DIA experiment, we can condense it down into like 30 to 50,000 MSMS spectra that are pseudo-MS spectra. And then we can check each one of those against a protein sequence database, and we make a peptide spectrum match, or a PSM, as we talked about earlier, based off of each spectrum, right? Okay, so this lets us say, okay, we've got 50,000 spectra that we're interested in, and so now we're trying to make 50,000 assignments for peptides in that experiment. The alternative approach here would be like taking all of the peptides in a FASTA database, say that 1 to 2 million peptides of interest, extracting out signal for all of those different peptides, and then determining what evidence is there for that detection. So this is interesting because it gives you the ability to say, well, there was no evidence for this particular peptide. That's something that you cannot do with a spectrum-centric analysis method. But it does have this major caveat where we're actually analyzing, again, in the top case, we are looking at 50,000 spectra. In the bottom case, we're actually looking at 1 to 2 million peptides. So what we're actually interested in is trying to ask, we're essentially asking 50 times more questions, which means we need to false discovery correct against a lot more types of peptides at the same analysis platform or same analysis set. Okay, so how do we think about building these types of libraries? Because you want to do this across a lot of different fragment ions, a lot of different peptides, across many different types of experiments. And so one way you can kind of think about this is using stable isotope labeled peptides. These can be any stable isotope labeled peptides or any peptides that you know that are not in your sample. So we talk oftentimes about IRT peptides, but you can also use other types of peptides. So for example, Pierce has a retention time standard that I frequently use. You can use BSA peptides. Honestly, you can use peptides that are actually endogenous in your sample just normally. But if you know an actual retention time for those particular peptides and you can map it to some index retention time, and again, here I'm using IRT as my index retention time, but it doesn't actually have to be IRTs. IRT is kind of like a dimensionless place that has no necessarily bearing in the real world. Like you can get negative IRT retention times, which makes no sense in the real world. So I can extract out those blue peptides out of my DDA run when I've injected them into, or when I put them into my sample and collected DDA. And then I can say for all of my other peptides that I've measured, I can map those to a specific point in that retention time line. So now I have a specific retention time IRT for all of those different peptides that I'm interested in monitoring. And so if I've got another type of peptide or another type of DDA run, maybe another fraction or something like that of the same type of sample or a different sample entirely, I can run a different DDA experiment, and it doesn't matter if those IRT peptides move around. They actually still give me enough anchors to be able to map to that same type of line. Now, Hugo asks, how many peptides do I need for our standard? Honestly, I think that the IRT standard is around 10 peptides. The PRTC is around 12 peptides. Something in that range is just fine. Digested, as I said, just taking enolase and digesting it, or taking albumin and digesting it, and then putting it into your sample at a certain small ratio is enough as well. So that's another good way to try to deal with this, as long as those peptides are not likely to be in your specific sample. So this is one way to think of it, and then you can map those retention times from one run to the next run. The other way to think of this is once you've actually mapped all those red peptides in that first DDA run to IRTs, now all of a sudden you actually have IRT measurements for all those peptides. So anytime you see those peptides again, they become your IRT experiment. So Hugo, thinking about this, you can actually use these now peptides, now that you've put IRT, now that you put heavy stable isotope labeled peptides into one sample, you can use any peptide that is assigned in that sample as now an IRT mapper. And so one type of approach that we published a couple of years ago takes those peptides and monitor it in one particular sample, and we map those to IRT samples. Now we can go back to all of our old data and we can say, well, we've got a bunch of the peptides because they're all within the same sort of framework. If I analyze, for example, in that red experiment, if I analyze and identify 5,000 peptides, now I have 5,000 anchors. And again, because I only need 10 of them in any given sample to be able to draw that line, I can now look at a green sample, totally doesn't have any IRT sample peptides in it, no stable isotope labeled peptides in it at all, but it has some of the red peptides in it. and I can look at a purple sample, and I can look at a blue sample, and I can combine a ton of data. So in this experiment that we did at this point 4 or 5 years ago now, we actually were looking at a swath of 1,000 different DDA runs that were acquired quite a long time ago, actually. This is basically our ability to take and mine all of those runs based off of a single DDA run that we ran, because we were able to assign anchors to all of those DDA, all the peptides identified on that single run, and then map all those new peptides into our new set of samples. So this is a great way to mine all the old data that exists in your lab, on your system, on your setups. That said, there are a lot of different other public available libraries. You can go to Swath Atlas, download some libraries. You can go to NIST and download libraries. That library that we talked about earlier, where we combined a bunch of data together, that was actually a phospholipid library linked into this library called Phosphopedia that we put up. So that's another great library source to be able to get access to for DIA or PRM types of experiments. So again, all of these are great sources. There's a growing list of these library sources, and I think you can definitely be excited about all those different types of sources, but be careful with them because they're not always ideal. You need to make sure that they're good for your type of experiment. So there are lots of different types of tools that are good for this type of experiment. We talked about a bunch of these, like Skyline. There are also other types of, for example, retention time-based filtering approaches like OpenSwath or Spectronaut or PeakView, some spectrum-centric approaches like DI-Umpire or PLGS, some RT agnostic approaches like Encyclopedia or Scaffold DIA, MS Split DIA, Pecan. There are tools that now split between these that take multiple types of approaches. So DIA neural network, DINN uses kind of a retention time-based filtering and then kind of thinks about it from a spectrum-centric perspective. There's Direct DIA from Spectronaut that kind of is a spectrum-centric tool as well. So there's like lots of different ways in which you can type of work with this, but they all they fall into these different brackets, but they all have different good and bad aspects to them. Again, the spectrum-centric ones all require precursors. The retention time filtering-based ones require really accurate retention times to be useful. The retention time agnostic ones are sort of more free, but at the same time become a little bit harder to judge FDR with. So there are lots of different kind of great advantages and disadvantages of each of these different quadrants. Okay, so why would we want to do this? One thing that we've noticed is that if we take a particular data set where we analyze it with PRM and we come up with good ratios for what a peptide specifically should be, we can be really confident that that type of measurement, that quantitative measurement is the right measurement. We can compare that with DIA methods. In this particular case, I've got 3 different DIA methods where I've got normal DIA, variable width DIA, overlap DIA, and we've got a DDA-based method. And one of the things that we find is that more of the time, we see that the DIA-based methods, they identify about the same number of peptides usually in this particular case, but they quantify much more accurately for DIA. So we get a ratio of what we measured versus what we expected to be as close to 1-to-1 as possible, whereas there's a little bit more scatter with the DDA measurement. And so one of the things that we see is that we see lots of these circumstances where if we look at sort of the width of that box over time, we find that the DDA actually just tends to introduce a lot more noise into the types of measurements that we're making. And one of the reasons for this has to do with the fact that with DDA, you are extremely reliant on getting an accurate monoisotopic measurement, +1 measurement, +2 measurement. If any one of those measurements is interfered with, then the overall signal that you're trying to work with is actually interfered with. So one of the advantages of DIA is that you can almost always get 3 good transitions out of any given peptide measurement. So in this particular case, over 80% of the detections that we're making here in this experiment have over 3 transitions. And because of that, because we've got 3 good measurements for those individual peptides, we actually are looking at CVs well below 20% on the vast majority of these cases. So we can be really comfortable that we're making good measurements in these cases because we can always throw out transitions. We're not reliant on being able to make measurements of we're not reliant on accurate measurements for everything that we measure. So for example, with DDA, you need the monoisotopic, you need that +1, the +2 to be able to integrate that peptide well. Here, we only need 3 transitions to work. Okay, so what are our potential pitfalls? I'm just going to go through this pretty quickly. One of those particular pitfalls is that oftentimes when we're looking at a particular peptide, we actually can see signal for that peptide that corresponds to that particular peptide from multiple different sources. What's going on in this particular case is I'm actually looking at a phosphopeptide experiment where a phosphate is actually being moved from one peptide to the other peptide, same sequence, but just different localizations of that peptide. And because there are different localizations, they move from one side, the retention time shifts considerably, and the relative abundance of the different fragment ions is a little bit different. Well, you get kind of 2 different sets of measurements. So what's going on here is if we're measuring, for example, these 2 CDK peptides, we've got a phosphopeptide where the tyrosine is phosphorylated down here and the threonine is phosphorylated up here. They have exactly the same parent masses, and the retention times may be a little bit different. But realistically, the only way that you can tell the difference between these is by using those specific fragment ions. So here, for example, we've got an isobaric peptide. So this is exactly the same mass, right? So both of these peptides have exactly the same mass, but they share a lot of the same orange peptides here. But there are some yellow peptide fragment ions and some blue fragment ions that are specific to one form versus the other, and that helps give you a chance to tell the sort of difference between these apart. And there are lots of different other types of modifications that can kind of cause different types of problems in these circumstances that you can look at. So for example, deamidation or oxidation all can sometimes exist within the same window. So for example, if I go here, I can, and I'm just going to skip a little slide just to make this a little bit more straightforward. So if I'm interested in analyzing this particular peptide and I'm looking at the modified, the sort of unmodified form and the oxidized modified form, they have very similar precursors. And if you look at all of the Y-ion fragment ions, they're all going to be exactly the same between these guys. So these red ions are going to be exactly the same between the only thing that you're going to be able to tell the difference on are these different B ions. And so when you look at these precursors, they actually exist within that same 24, 25 MZ white isolation window. And so you're co-fragmenting them at the same time. And so if you actually try to do extraction of these particular peptides out at the same time, you get essentially the same signals. And so you might actually end up mistaking one for the other. And so this becomes a potential pitfall. So this is one of the reasons why we don't necessarily recommend searching for modifications in a lot of these types of searches. Or if you're searching for modifications, be very sort of savvy about what your precursor isolation window widths are and that sort of thing. Okay. So let's see, this is a good point to start. So Hugo asks, should we discard less than 3 transitions with DIA quant? Oftentimes, I usually require 3 transitions. I think this is a really good way to go. But you can work with fewer transitions. You just have to know that your signal, your measurements are actually just not as good or not as accurate. So typically, I require 3. Oftentimes, I actually will require even more than that. And Lima asks, for IRTs, could I use a digestive standard of enolase? Absolutely. Yeah. So that's what I'm saying is as long as it's not a protein that you expect to see in your sample and you're not trying to measure in your sample, you can absolutely use it. Also, if you know a retention time standard for any given peptide in your sample, you can use that as the type of measurement. Sumit asks, can you use something like iTrack with DIA? Reasonably, you cannot actually. iTrack and DIA are pretty much not. You can use something like complementary TMT potentially, theoretically, but it's just not a very, it's not likely to be, there's a lot of research that needs to go into that type of work before you actually could do something like that. You definitely can't use something like normal eye track because of all of the quantitative information is essentially lost because you're actively seeing co-eluting isobaric peptides at the same time. Okay, so let's talk about briefly best practices for, before we get there, Leanne, am I saying that DIA is not great for looking at PTMs? I think actually DIA is one of the best ways to look at PTMs, but you have to be really savvy about what types of PTMs you're measuring, and you have to be able to use localization search engines. And we publish some of those as well. So with an approach called Thesaurus that we are really excited about. Where we actually see a lot of faults of doing DDA analysis. In fact, from our perspective, we think that you can't really analyze, you can't get robust information about phospholipeptides from DDA based off of the information that we've learned from analyzing phospholipeptides with DIA. So we think that the DIA is a perfect tool for being able to analyze phospholipeptides, but you have to be smart about how you analyze them. You have to use the right software. You have to think about it a lot. Okay, so best practices for setting up DIA, and then we're going to talk about building DIA-only library data sets because they kind of go hand in hand. Because at this point, from my perspective, actually, I have stopped acquiring DDA-based libraries. Most of my work now uses DIA-only libraries because I find it to be just as accurate and considerably faster for the work that I am interested in doing. So to understand how you set up a DIA experiment, you actually have to go back to think about how an instrument actually works. A lot of the DIA experiments were originally designed for TOFs because TOFs actually have a lot of the same types of characteristics as triple quads. So one kind of advantage here is that that basically you rather than having a QQQ, you actually have a QQ TOF, right? And so here, essentially all you're doing is you're taking peptides that you're interested in, you select out a specific mass over charge range, you fragment those ions in the middle quadrupole, and then you push them into the TOF. And so the TOF pushes them down and they hit the detector. And the advantage of this is that you get measurements that are very, very fast. So thousands and thousands of pushes per second. And the other advantage of this is you get real scans, real profile scans for every different type of peptide. So you're always going to see signal after you've analyzed enough of these pushes. But one kind of disadvantage of this approach is that the pushes happen so fast that that you actually aren't looking at that many ions at any given time point. So if you're thinking about what you're looking at a millihertz kind of scan, so if you're looking at 1 in 1,000 1 over 1,000 seconds to accumulate ions and put them into the TOF. That's not very many ions, realistically. Okay, so a lot of thinking about the original sort of swath paper that was interested in analyzing all peptides from 400 to 425 across this mass range. So they built out 32 windows that were 25 MZ wide. One of the things that they were pretty clever about early on was thinking about the sort of distribution of peptides that they're actually being able to see. Most peptides actually exist in this 400 to 1,000 range here, and there are fewer and fewer peptides up above that. So you can use essentially what are called variable width windows across that time point. And so the earliest approaches use 32 windows and then showed the variable windows where you have wider windows, the higher MZ range where you have fewer peptides that are co-eluting at the same time, and narrower windows at the low range work pretty well. But 32 windows, because the TOF can scan so fast, they thought that you might as well try to sort of push this. So 30 windows became 40 windows, became 60 windows, became 120 windows. And so this is kind of like the current standard now, where your minimum window size for the windows at the middle are around 5 MZ wide windows. And they get out to around 40 MZ at the top range. But that best range is in that 5 MZ range. So an approach that people have been doing, remember how I talked about how transmission efficiency on the window edges is not as good. One way to try to deal with that is by just tacking on margins under the side of those windows. So this is, I think, a very good way of analyzing data using a TOF. Now, I want to contrast. So this is sort of the common practice that has been pushed by, for example, SciEx. And I think this is a great way to try to do this type of work. Okay, so that said, there are very different constraints when you're thinking about this with an Orbitrap. So if you're analyzing data with an Orbitrap and trying to do DIA here, you end up with an Orbitrap, which is actually a relatively slow mass measuring device. So instead of being able to analyze thousands and thousands of scans per second, you're actually only analyze maybe 10 to 20 scans per second.
Brian,
so
there's a question about TOF. So how much scan time should I set, or at least a reasonable time? 0.3, 0.5, 1 second?
Oh, again, that really depends a lot on the specific TOF that you're actually working with. That one, I think, is worth talking to the specific vendor. So if you're working with SciEx or Agilent, you should talk to the vendor about how you should configure the instrument on those specific TOFs. Again, remember the kind of constraints that we've got where you have a need to be able to make the same measurement every 2.5 or 3 seconds wide. So Sean asks, would a TIMS TOF setup be similar to a SciEx TOF? And actually, it's very different. A TIMS TOF, because of the constraints of having the ion mobility cell up front, you actually have very different ways of acquiring that data. And so all of the TIMS TOF DIA approaches so far have actually gone back to the original 25 MZ wide windows across the same range approach because of the need of making everything work within the ion mobility space of the TIMS device. Okay, so again, an Orbitrap is a relatively slow device. So if we're thinking about trying to make measurements across that same type of window, you know, at best, we're looking at 25 windows in that same range. We are actually looking at very, very wide variable width windows. So we're talking about 20 MZ wide windows in the best part and up to 150 MZ wide windows up at the long part. So this actually is kind of a terrible way to use a TOF. An Orbitrap, is to basically take the ideas that were developed for TOF instruments and apply them here. We really need to develop new types of ideas here. Okay, so there are some pretty big advantages in trying to do this with a TOF. And I want to just sort of brush through these really quickly to give you kind of an idea. One of them is that we really kind of can think about limiting the mass range a little bit more. So historically, we've only been looking at from 400 to 1200. And really right here, actually, we could focus in on this range from 400 to 1000. And we have some data that actually backs up this idea of saying that we get the most number of detections from this 400 to 1000 different window set for making these types of measurements with an Orbitrap, and that range seems like it's the best type of range for our types of experiments. But if you're looking at phosphopeptides, you actually kind of would want to have a wider window or a different type of window. So maybe you would consider from 500 to 1100, for example. Another approach that I think is a really powerful approach is essentially an algorithmic way of doubling your resolution with the instrument. And so if we think about this type of approach where we've got precursor scans and Bragmanine scans one after another, and they've got window boundaries as shown here, well, we can think about, well, what if we shifted over every other scan, every other set of scans for cycle by 50%. And this goes back to that first question about, well, whether we could analyze overlapping scan windows, and that we can actually use this to our advantage. So if we have overlapping scan windows, or in this case, we like to call it staggered windows. So if you're looking at an MS2, for example, here, all of those ions in black for that given MS2, here is a particular window that you're looking at. And if you look at the windows before, immediately before and immediately after, some ions are going to be present in both of those cases. So these red ions you aren't going to be able to say much about. But if you find ions only in the early part, so in this particular case, if we're looking at our center window is from 600 to 624, and our previous window set is from 588 to 612, we see that there are ions in green. And these ions in green, we can say, are definitely only in the left side and not in the right side. And so we can actually assign those fragment ions only to a scan that belongs from this 600 to 612 range, because they have to exist in that first window and not at all in the second window. And so we can do the same thing on the other side, where any ions that are associated with the previous and next scans on the right side can be associated with a new spectrum, a pseudospectrum that is associated only with the range from 612 to 624. And so this is a really great way of simplifying our experiments and being able to essentially increase the speed at which we're acquiring data by 2 times without having to do any actual adjustments to the instrument. I mean, think about this. If Thermo were to come out and say, we've got an instrument that collects Orbitrap scans twice as fast, you'd be like, wow, that's amazing. Here, we're actually able to do that algorithmically without even having to change the instrument. You could do this on any type of instrument that Thermo's made for this. That said, staggering is incompatible with this idea about putting margins on the side. So we have to come up with a different strategy for dealing with that type of thing. And this is where we think about what we call forbidden zones. Forbidden zones take advantage of the fact that peptides are made of the same stuff. They're all made of hydrogen, carbon, nitrogen, oxygen, sulfur. And because they're made of the same stuff, they only can have certain precursor masses. So there are certain types of masses that they just cannot take because they are not made of weird atomic, different types of heavy metals or different types of other atoms. So those spaces between where you expect to see peptides are where we can put what we call optimal boundaries. And these optimal boundaries are reasonably wide. So for example, if I look at, and they seem relatively straightforward. So when I think of the boundaries that I would look at for Swissprot and map those to the boundaries that are where I see peptides, and looking at all the peptides of Swissprot and then all the peptides in the pan-human library, they actually overlap quite a bit. And they fall in these kind of peaks where you expect, so any peptide that has a +1, a +2, or +3 charge falls in this particular high peak. +4s are in the shoulder, +3s are in this peak here, +2s and 4s are in this peak here, +3s are here, +4s are in the shoulder. And again, you get to the next monoisotopic peak where you get +1s again. So this is where you expect to find peptides, where you see these signals. So it's very easy just to stick those boundaries in a region where you don't expect to see peptides. That said, other types of peptides have different forbidden zones based off of their chemical characteristics. So you have to compute these separately. So for example, phospholipeptides have a slightly shifted forbidden zone that we've seen based off of different libraries, and other types of modifications have different forbidden zones. So if you try to do this type of work, it's worth trying to think about the math for where you would draw these boundaries. Okay. A final sort of approach that we like to take is this sort of idea of gas phase fractionation. That's where, rather than trying to analyze an entire acquisition or entire sample within one acquisition, so from 400 to 1,000, we're actually interested in just saying, well, what if we injected it twice? And instead of trying to just do it twice like technical replicates like you would do for DDA, why don't we just make the first measurement from 400 to 700 and the second measurement from 700 to 1,000. This gives us much narrower windows and gives us better isolation and better characteristics of being able to identify peptides. So if you have the ability to spend multiple acquisitions, we find that this is a very accurate, very good way of dealing with this, as described originally by Dave Goodlett's lab in the early in 2008, 2009, and again, sort of reused in this particular case for modern DIA in Sonia Ting's paper in 2017. And we've got on our website, and again, you have access to these slides, so you don't need to write down these crazy long URLs, but we've got lists of parameters that we recommend for how you would set up these types of experiments based on, as well as the window widths, based on the sort of constraints that we've talked about so far. So what you're going to set your AGC targets for and your max ion inject times and those types of things that you would want. Okay. Again, I would also recommend referring to this paper that I put into the Google Doc, but also just to read through it to kind of get a refresher on this type of stuff. We've also got a quick start guide for how to deal with this. Again, that same recommendations for DIA settings on Orbitraps. So in the last just sort of couple minutes, I want to talk very briefly about this idea about building libraries for DIA-only data sets. And this is something that's actually been really important in our lab that we've been moving forward on. And the reason why is because DDA libraries are, they're very expensive to make, right? So if you're interested in analyzing a specific sample, say HeLa or MCF7 cells or something like that, or yeast or E. coli, you really need to acquire tens of samples tens of injections of fractionation to be able to analyze that, to be able to representative library for that particular sample. And one of the things that actually explodes if you're trying to monitor all of or detect all of the possible peptides for that organism. These experiments take a lot of effort, a lot of time. And it gets even worse, actually. If you're trying to look at the overlap between different types of fractionation methods, they actually identify very different types of peptides. So if you're using SEX or high pH reverse phase to do your fractionation to build libraries, you identify very different types of peptides. So in those particular cases, because those different types of peptides identify, different blocks, you actually kind of need to use multiple types of fractionation methods to build those peptides. The other aspect of this is that collision energy optimizations are actually a little bit off. So for example, as we talked about originally, DIA is not really perfectly set up for being able to make collision energy or the fragmentation parameters for DIA are not exactly the same as for DDA. And as a result, if you're looking at Pearson correlation, as I'm showing at the bottom, most peptides have good Pearson correlation if they're +2 peptides, but +3 peptides tend to do a little bit worse, and +4 peptides tend to have very bad correlation relative to what you see with DDA. I think the final and most kind of weirdest aspect of this is that you actually see matrix effects caused by fragmentation, by sort of fractionation methods. So if you're analyzing a sample with a much lower matrix than your actual samples are, then you get retention time shifts because there are different types of peptides that any peptide you're interested in is going to be interacting with on that column. So you end up with sort of these time shifts in weird areas, and you get shifting of different peptides backwards and forwards because of the fact that they have a simplified matrix associated with them. So that even if you're using lots of different fractionation methods, you still get inaccurate retention times because of this. So we've actually been adopting this kind of approach of trying to do library prediction. In this particular case, we're using PROSIT quite a lot. And this is a tool that we recommend. And we've developed tools and workflows around this in sort of inside of Encyclopedia, where there are mechanisms to take a FASTA database and convert it into a PROSIT input so that you can stick that up on a PROSIT website, and then analyze those data, bring those back into Encyclopedia, build libraries for them for DIA, and be able to analyze data with them. And we find that the predictions are actually really shockingly accurate. So for example, this is a particular peptide. I pulled this one at random, where you can see the fragmentation patterns of this particular peptide are almost exactly on. They're not always perfect, but they are almost always like within a very reasonable error margin. And the retention time prediction based off of the deep learning model in PROSIT is off in this particular case by maybe 1.5 minutes, but that's actually about as accurate as a DDA experiment would be with fractionation. So to kind of give you an idea, basically PROSIT is predicting libraries that are as accurate as DDA-based libraries, is kind of our take-home based message on this. But still, when we're analyzing data with these DDA libraries, we actually still see more peptides, more proteins than we do with these predicted libraries. And again, these here we're making a combined SEX, HPH, a high pH reverse phase DDA-based library. So in analyzing yeast, we're looking at 16 fractions here, and analyzing HeLa, we're looking at 39 fractions. We're identifying a little bit more than we do with the predicted library. And again, this has to do with the fact that we're analyzing all the tryptic peptides in the FASTA database. And there's just too many peptides for us to look at. And so one of those aspects is we really need to look at as small a database as possible. Again, don't consider PTMs unless your experiment is about PTMs and you have enriched for those specific PTMs. But we've started to work on this approach of trying to make it a little bit easier to analyze this type of data, where instead of trying to think about gas phase fractionation for 2 acquisitions, we actually have increased this a little bit more, where we're actually doing 6 different fractions associated over time. So we're doing 1 fraction acquisition, a second acquisition, so 6 injections for this sample, in which we can analyze the sample with a huge degree of accuracy, with basically, essentially, very narrow precursor isolation windows. And so this is kind of like the new workflow that we've been using, where we take these sort of samples that we acquire from our different pools. So we're basically taking a pool of this yellow sample from our blue samples and our orange samples. We're injecting that yellow sample 6 times using gas phase fractionation. We're searching that using the PROSIT predicted library. And then we're making here a library that is associated with all of the possible peptides that we can see in this sample. So now we've identified all the peptides that we're confident in seeing in either the orange or the blue samples. And now we could just take our quantitative samples and then look those guys up very accurately. And the advantage of this approach is that we actually get extremely accurate retention times in this approach called the chromatogram library approach. So instead of looking at plus or minus 5-minute windows, we're talking about plus or minus 20-second windows, because we know exactly where that retention time should be. And our fragmentation, because it's acquired with DIA, we're actually getting really good DIA-based measurements here. So finally, we see with this type of approach, we actually are getting about the same number of detections as we do with these very large libraries. So for example, if we can use this empirical correction, this chromatogram library and correction, to improve any type of detection approach. So in this particular case, we're looking at here, if we're looking at the DDA libraries, we can actually build libraries that actually help us identify more peptides. And then we can do the same thing with these predicted libraries. But I think the biggest advantage here is that now we essentially make the empirical corrected DDA libraries essentially the same as the empirical corrected predicted libraries. So this enables us to build like a DIA only workflow that it allows us to be able to identify every type of peptide. So with this, I'd just like to close with a couple of recommendations. DIA is a fantastic tool. It's not the end-all be-all for every type of experiment, but if you just need a quick answer and you need kind of like, you've got a really simple matrix, then DDA can be a really great tool. But there are different types of approaches that I would recommend, for example, using gas phase fractionation if you have only a handful of samples. If you've got a lot of samples, then I recommend building these gas phase fractionation libraries, these chromatogram libraries, again, using predicted libraries, and then using DIA, a single injection DIA to analyze your quantitative samples. PRM is sort of the same workflow, or sorry, with PTMs, it's the same workflow, except there I would highly recommend using a DDA-based library because the prediction algorithms aren't very good right now for modified peptides. So with that, I'd like to finish, and I'd be happy to stick around in the break. I know we've gone over a little bit, so I'll stick around in the time between Lindsay's art session and then Lindsay's session and answer a bunch of these questions as we go.
Brian, thank you so very much. We have a virtual round of applause for Brian. Thank you for this comprehensive overview. There are quite a few questions left in the Q&A, and there's also one question for you in the chat at the very bottom of the chat as well. So for everyone else, we have a break until 12:45, so we can use this time to go over questions or just stretch your legs, and we will be back at 12:45 Eastern for Lindsay's presentation.
Great. So I'll just kind of like go through these a bit. So for example, the one issue with the staggered window scheme. That's the overlapping window scheme that I talked a little bit about. Let's see back here. So this approach here, I said that this approach here isn't compatible with window margins. It's also not compatible with variable width windows too. So that is kind of a limitation. But there are kind of other interesting ways in which you would want to think about this from a TOF perspective. So David Schmidt brings up a really interesting point about what about scanning swath methods? That's where you actually take the quadrupole and you sort of scan it across time. And actually Waters came up with the first implementation of this called Sonar. So Sonar came out a couple of years before scanning swath they're essentially the same type of approach where you're scanning the quadrupole over a specific window and you're essentially acquiring micro scans at each of those different windows, and you can use linear algebra to be able to deconvolute those together. It's actually a lot of the same type of math that we're doing here with these fixed scans with the Orbitrap. You just can't do a scanning Orbitrap essentially because the Orbitrap is too slow. So those are great questions. So when you're interested, Gayatri asks about using SIL peptides or stable isotope labeled peptides for doing DIA analysis with Encyclopedia. So right now the only way to do to integrate those with Encyclopedia is to actually have them in your library as well. So you can definitely do the integrations for them, but you treat them as independent detections and you interpret them on their own. So for example, if you've got your normal library, you can add in the stable isotope labeled library or for those specific peptides at least. So that's a fantastic question. There's actually new tooling in the new 1.2.2 to be able to deal with that type of library addition. Okay, so can I elaborate a little bit more on the GPF strategy for building libraries? And I want to show you this slide here again. Okay, so here again, so we've got 30 samples. Say we've got 30 samples and we've got 15 blue samples, 15 orange samples. We want to analyze these together and we want to build a library for them. So what I will do in this type of experiment is for these 30 samples, I will take a sub-aliquot off the top of each one. And oftentimes you can get a microliter out of the bottom of each well anyway. So this is free sample when you think about it. So I will take a sub-aliquot off of the bottom of each one of those, pool them together into a single pool. That's that yellow sample. And then this yellow sample, this pool, I will analyze this with a different framework. So the orange and blue samples, I will analyze those for quantitation. And the yellow sample, I will analyze for detection. I want to detect every possible peptide in that sample as accurately as possible. So I do this sort of 6 gas phase fractionation method where I acquire 2 MZ wide windows across the entire window range, 400 to 1,000. This is basically like doing PRM, right? PRM isolation is about 2 MZ wide windows, but it's PRM for every peptide in the proteome. You can't do this for every sample, right? That would be way too much work to do 6 injections for every sample. But you can do it for a pool very efficiently. So in this particular case, I would acquire my 30 orange and blue samples and my 6 gas phase fractions for my pool. And so now I've got 6 injections of PRM quality data. I detect all of the possible peptides out of this data set. So again, 2 MZ wide isolation means that this is as good as DDA based isolation. So you're seeing as those types of peptides in the same so you're seeing them with the same type of isolation that you would with DDA, except you're actually able to see the apex of the peak, not just the shoulder of the peak for collecting the MS2s. They're just much deeper. Typically in these gas phase fractionation libraries, we can identify tens of thousands of so oftentimes we can see up to 100,000 different peptides that we can confidently identify. And once we found them in the pool, Well, now we know the exact fragmentation patterns and the exact retention time standards for those specific peptides. We can map those to the rest of the experiment. So for example, I can say, well, I found this peptide that I'm interested in. I know exactly where it is in time and space, and I can go look it up with the right tools. So we've tried to make this workflow very easy to use in Encyclopedia, but you can use this with other types of methods too. We've done it with Skyline, for example, before. Okay, great. So Charlie Yang asked the question about IRT peptides. IRT peptides are just a trademarked peptide type that fit within index retention time. These are peptides that are produced by a specific company, Biognosys. So they're sold by that company, and that's fine. But any type of peptide works here. So pure retention time standards, PRTCs, are essentially the same thing. They're different peptide sequences, but we know where they should be on the retention time scale, the same IRT scale even. And so you can actually use those as your retention time standards as well. We also know the retention time standards for many of the different peptides in the human proteome already. So we know what their appropriate IRTs ought to be. And so you can use just those peptides as your retention time standards are as well. We're actually at the point now where realistically, in very few circumstances do you actually need heavy labeled isotope peptides to be able to use as retention time standards. You oftentimes need to use them as quantitative standards, but in certain circumstances, but that's not really what IRTs are about anyway, right? We're just focused on monitoring retention time in these cases. So here for retention time alignment, you know, we're at the point now where you don't really even need to consider adding spiked in peptides into your samples, unless you, for some reason, need to monitor those types of peptides for another type of reason. So again, these types of peptides you don't even need to necessarily include them. But if you do put them in, I recommend putting them into whichever samples you're interested in monitoring. So put them into the same sample, injected into the same sample, so you have a certain amount of them. Essentially, this can be a nice kind of online quality control if you have these peptides around. Again, they're not necessary, it's just if you have them around. So is there a difference between gas phase fractionation and DIA and just DIA from Susanna? This is a fantastic question. It really is just a limited retention time window. Mass range, as you say. It's really just taking the mass range. Instead of going from 400 to 1,000, you're going from 400 to 500. And the second injection, you're going from 500 to 600, 600 to 700. Everything is exactly the same. That's what makes it great for building libraries, because all your fragmentation patterns are exactly the same. It's the same matrix, so all your retention times are very precise. So this becomes a much more popular, practical way of doing library generation, at least in my mind. And then I've essentially stopped building DDA-based libraries. While they're oftentimes used by a lot of the other types of search engines, this type of DIA-only library generation I find to be very practical for virtually every type of experiment that I do that's DIA-based. So is there any type of software that we need for doing gas phase fractionated DIA? Encyclopedia, the tool that our lab creates, actually has all of the GFP DIA analysis stuff built into it. You can do some of this work in Skyline, and that's something you can ask Lindsay about afterwards. But it is possible to do some of that work in Skyline. But we recommend oftentimes just building, even if you're going to go down that route, building the libraries inside of Encyclopedia and then exporting them for Skyline, because the tooling there is just designed to create these libraries from gas phase fractionated samples. Marco asks a fantastic question, which is, if you're building from hundreds of samples at the same time, is it too diluted at this point? for any given peptide that you're interested in measuring. And if your peptide only exists in one of your samples, then yes, it's probably too diluted. But that peptide is probably not very useful in terms of trying to do quantitative analysis across all of your different runs if it only exists in one biological replicate in one of your samples. It's just not going to be statistically useful going forward. So really, this is actually enriching for peptides that are likely to be statistically useful in your experiment going forward. That said, if you have two very disparate types of experiments, so for example, one experiment I was doing was looking at CSF from two different types of sources. So one is from a lumbar source and one is from ventricular source. These are whether you collect it from the base of the spine or whether you collect it from the back of the head. And there, because of the sort of difference in the type of matrices that those things have, they have very different types of proteins on them, whether it's from the spine or from the back of the head or the spine. So because of that, we actually oftentimes will acquire two different libraries, one for the lumbars and one for the ventriculars. So that's a fantastic question. And we'll analyze the ventriculars as one block and the lumbars as another block. So if you've got two very different types of samples, so for example, if you're trying to compare two different cell lines or something like that, then that's a great way to go.
So unfortunately, we're out of time. But Brian, do you mind staying around a little bit and answer the questions in writing?
Yeah,
you
so
can
absolutely
type the questions while we start already the next part of the program.
Yep, and I'll stick around through Lindsay's as well.
Thank you so very much. We really, really appreciate your presentation. And so now it is time for me to introduce our second speaker, and I'm really excited that Lindsay is joining us this year. So Lindsay was trained at the Broad Institute working in targeted proteomics, and then she completed her PhD in the same lab as Brian at the University of Washington in the lab of Mike McCoss. And currently Lindsay, a postdoctoral fellow at University of Pennsylvania. And Lindsay also has a lot of achievements as an educator. She is a veteran presenter in the Skyline short courses and also at the May Institute. And we are very happy that Lindsay could join us again this year. And I forgot to mention that both Brian and Lindsay have been named rising stars in proteomics and metabolomics by Journal of Proteome Research this year. So help me welcome Lindsay for the second part of the program.
I'm super excited to be here again, even though we can't be up in Boston. It's always a fun crowd, and following Brian up is definitely a challenge, but we're going to try to give you a little bit more in-depth discussion about some of the things that Brian talked about. And especially I'm going to be trying to frame these within this software tool called Skyline. So we're going to have some hands-on with Skyline later. Try to download those files if you haven't already. And hopefully the TAs can help you get set up if you're having issues installing Skyline. I also have a couple disclosures. As Olga just said, I'm currently a postdoc at the University of Pennsylvania, but I also have co-founded and currently consulting at a company called Talis Bioscience. So as I had mentioned, I kind of have three main objectives for everyone today. So the first, I want to revisit some of those core concepts that Brian gave you a theoretical framework for this morning. And I want to reframe them using the Skyline ecosystem. So the two kind of main things here are going to be developing DIA assays and visualizing DIA data. I want to give us a little bit of a deeper understanding for quantitative liquid chromatography mass spectrometry data, including how chromatogram peaks are processed in Skyline. So Brian talked a little bit about how the data can be analyzed. We're going to try to give you a little bit more background on that so that you have a better intuition for what this black box is doing with your data. And then throughout, I'm going to have a lot of deeper dives, a lot of references for you to follow up on, including things like Brian talked a lot about this idea of index retention time standards, retention time alignment. I'm going to throw a whole bunch of references for you to dive a little deeper if that's something that you're interested in. So throughout the talk, the first part of the talk, I'm going to have anything that's relevant to Skyline as the software is going to have this kind of blue background with the white text. Just to orient you really quick, and if you'd like, you can follow along with Skyline open on a second monitor, or this playing in the background while you look at Skyline. But kind of the basic structure of the Skyline program when you open it, on the left-hand side, there's a window called Targets. That's where your proteins, your peptides, and your fragments are going to be. Up in the top right corner, you see this kind of squiggle, swirly blue thing. That's supposed to be a peptide secondary structure, an alpha helix. If you drop that down, you may notice that there's two other settings for Skyline. There's also a small molecule setting, and there's a mixed setting for peptides and small molecules. And then down at the bottom, in the bottom right, there's a series of target summaries. So the number of proteins, peptides, precursors, and transitions that are in your document. So we're going to come back to Skyline throughout the lecture portion of this, and then we're going to be opening up Skyline and playing with it ourselves later. But what Skyline does is it's this ecosystem for various informatics used in quantitative mass spectrometry. So when we have a mass spectrometry question that we want to ask, a proteomics question, usually we start with some sort of hypothesis. In a targeted setting, your hypothesis might be a specific protein of interest or a specific pathway of proteins of interest. In DIA, as Brian just told us, what's really nice compared to a PRM, a targeted study, is that you don't necessarily need to know what question you're asking when you acquire your data. Skyline also assists in the assay development section. As Brian had mentioned, if you're doing targeted, like a PRM, there's a lot of upfront assay development that you have to do for those specific proteins of interest. For DIA, there's a little bit less assay development upfront, but there are certain things that we do have to decide, like Brian talked a lot about different isolation window schemes that are going to makes sense depending on what kind of instrument you have and what kind of samples you're working with. The scope of the experiment might dictate the kind of isolation window scheme that you're optimizing. Then we talked a lot about spectral libraries. So that's another thing that we can do in Skyline. We can make the spectral libraries. We're going to be talking a little bit about that. And Skyline won't tell you how to do the acquisition on your instrument. You have to set that up, but you can actually export methods from Skyline. Methods directly from Skyline. It can analyze your data, process it. There's also several core analyses built into Skyline that we're going to talk a bit about, and even post-hoc additional analyses, post-processing, using various tools, including there's an external tool option for MS Stats, or you can export your data from Skyline, bring that into the MS Stats R package, and perform any of those R functions based on the input that you got from Skyline. So Brian gave us this good overview, right? When we're doing proteomics, we start with some sort of tissue or biofluid that we're interested in. We take the proteins out of that system. We perform the enzymatic digestion. We end up with peptides for mass spectrometry. They're separated by liquid chromatography. They go through analysis on the instrument. So precursors are selected in a targeted analysis. That's a very specific precursor. In DIA, we widen out so we get a whole range of precursors. Measure the fragments. We end up with mass spectra from the instrument, but we can stack those mass spectra up into chromatograms. So let's focus on this top part first, right? So there's a lot of different ways that we can get our protein. If we're going from tissue, we might have to perform processes like tissue homogenization. If we're in cell culture, we have to lyse our cells. We may decide to do further processing like subcellular fractionation. Now we have proteins. We may further fractionate our proteins, and Isabelle Bludau is going to give us a little bit of a vignette on some interesting protein extraction processes called size exclusion chromatography to get at protein complexes. As Brian mentioned, we can do PTM enrichments. We can do immunoprecipitations for specific proteins. For enzymatic digestion, we have a whole nother set of assay development questions. What kind of enzyme are we going to choose? Trypsin is very common. And as Brian mentioned, if we're doing DIA, most of these analysis tools assume you're using trypsin. But there's a lot of other options out there. Or you may be doing things like immunopeptidomics, which don't need to be enzymatically digested. And now we have peptides, but they're not quite ready for mass spectrometry yet. We might have to do more enrichment, another round of PTM enrichment. We might have to do another round of desalting. So this is just cleaning up the peptides. A lot of times these upstream processes either involve detergents or salt that need to be removed before we can analyze them at mass spectrometry. At the end of the day, we end up with this set of peptides, right? And in a targeted environment, you know a lot of these details. You know a lot of these details about the mass spectrometry prep. One of the first things that we have to do in Skyline is record some of these method sample preparation details in there. So Skyline, really the core of Skyline is making sure you have these settings correct. So some of the major setting features here. So this is up in the top left-hand side of your Skyline document. There's a settings tab. And if you go to settings, peptide settings, you'll see a window, pop-up window looks like this. It has multiple tabs here, things like digestion, prediction, filter, library, modifications, then finally quantification. So the very first thing, no matter what you're doing in Skyline, whether it's DIA or a targeted analysis, you want to set up the digestion and the filter. So some of these things, for example, the enzyme, you know that from just whatever you did in your sample prep. In the filter, in targeted settings, typically just using default settings in Skyline is perfectly fine. There's also under settings, there's settings peptide settings, and there's also settings transition settings. So here again, there's going to be a filter tab that we need to set up for targeted experiments. When we're doing DIA experiments, there's a whole nother set of settings, and Brendan's going to be going through these in much more depth when we do a DIA data analysis with him tomorrow.
Lindsay, there's a question. Can you add multiple enzymes if you're using a combo of trypsin plus something else?
Yeah, so you cannot add multiple enzymes. So it's just a drop down. You can't like check multiple boxes for multiple enzymes. But what we can do is you can create your own enzyme and just give Skyline the rules for your kind of like custom enzyme. So you can kind of not hack Skyline, but a little bit. It's a hack to make it accept multiple enzymes. That was a good question. Yeah, so we're going to be looking at all of these settings ourselves in depth in just a bit. But the final thing that I wanted to mention in these settings is under the settings transition settings, under the full scan tab, there's this MS filtering section kind of in the middle. If you set the acquisition method to DIA and you go to the isolation scheme, you have the option to add an isolation scheme in Skyline. And what that allows you to do is you can calculate your own isolation schemes using Skyline's isolation scheme calculator. So this is another option in addition to using the encyclopedia software Brian mentioned, also has an isolation window scheme calculator in it. This is just another option that'll let you do that. So I just kind of plugged in some of the options here. For example, Brian talked about doing that 400 to 1000 MZ range. We can plug in a 24 MZ window width. He talked about optimizing the window placement for those forbidden zones and how that differs for different types of samples, but you can check that box and it'll optimize the window placement for a whole proteome, not a phosphoproteome, just a regular proteome digest. The deconvolution, you can select the stagger, you can select the margins, just like he talked about before. And what that'll do is it'll populate this scheme here, which you can then make graphs for, or you could copy that into your instrument itself. So that's just a little bit of background, putting some of the things that Brian said into context, but I wanted to dive a little deeper now into the analyzing of the data. So now let's assume you filled out this windowing scheme or you followed the recommendations Brian gave you, you went to your instrument, you acquired your data. Now let's talk about processing that data in Skyline. So what's really nice, Skyline is vendor neutral. So it's going to take your raw spectra and you can process it all directly within Skyline. Brian also mentioned there's options where you can combine other software and pull that into Skyline. We'll talk a little bit about that. But I wanted to give you a bit of background into what exactly Skyline is doing with your data. So there's several steps when Skyline's reading in your data, and we'll again be talking about this more in depth tomorrow. But the first step, Skyline is extracting chromatograms. Then it resamples the chromatograms, looks for where peaks might be in your data, groups them together, and then performs the peptide identification. So a little bit more on those. So very first thing, as we just heard, mass spectrometry data is three-dimensional, right? So we have a retention time dimension. This is the peptides coming off of the liquid chromatography column. Then we measure the mass to charge for all of those peptides as they come up off the column. And then finally, we have a measure of intensity. So when we look at that overall, we get this kind of three-dimensional representation of our mass spectrometry data. If we take one set retention time and draw the slice of mass to charge and intensity data for that one retention time, we get the mass spectra. Conversely, if we hold the mass to charge set at one value and then extract the intensity across the retention time, we get the chromatogram. So Skyline's not thinking about the mass spectra, it's thinking about the chromatograms. So everything that Skyline's doing is kind of in that chromatogram space. So we set a mass to charge at a given value, we extract the intensity for that mass to charge over all of the retention time that we measured. We have a chromatogram extracted, an extracted ion chromatogram, XIC. So for the chromatogram, you might notice that I've drawn these small dots spaced along this black line. That's actually where the mass spectrometer made measurements. So each one of these points is one MS-MS spectra. And all of our mass spectrometry data, these are not spaced entirely perfectly apart. It's not a perfect sampling. So one of the first things that Skyline's going to do is look at all these scans made by the instrument and resample it. So this is because downstream processes in Skyline, the scans have to be on an even interval. So Skyline's going to look at the data, it calculates an interval width that best fits your data. Does this using something called a linear interpolation. But it's going to look at the data and say, if I sample at this interval, I have captured the shape of the chromatogram and I haven't distorted the data. If the intervals are too wide, the shape of the peak would become distorted. If it's too narrow, then the processing time will get jacked up. It'll take a long time to process the data. So Skyline's going to look, it's going to interpolate using that linear interpolation, a resampling across these peaks. Now it's going to look through the chromatogram and try to determine where the peaks are. So Skyline uses a peak detection algorithm that's called Crawdad. It's based loosely off of another previous McCoss Lab grad student, Greg Finney. Crawdad looks at the data, it finds the minima and the maxima across the chromatogram. So I'm kind of using these red dots to symbolize it's looking for the minimum, it's looking for the maximum, and then finally it looks for the inflection point, kind of the middle of that peak. So that's where the second derivative is equal to 0. If you remember back to your calc or pre-calc days, it finds minimum, it finds maximum, it finds the inflection point in the curve using the second derivative equal to 0. These set of points, minimum, maximum, and the middle, defines the detected peak. So you can kind of see this in Skyline a little bit. In Skyline, in the chromatogram window, if you right click and you go to transform, you can choose the second derivative transformation in your data. So Crawdad's placing those peak boundaries in Skyline. It's going to set them initially at those inflection points. So remember the inflection points where the minima is, where the second derivative is 0. So that's what we're showing here. We're showing the second derivative of your chromatogram. It looks beautiful. Where the second derivative is 0, that's the inflection point in your peak. Then Skyline moves those out a little bit because we don't want the middle of the peak, we want kind of the base. Okay, so now Skyline has gone through your data. It has found places that look like peaks using this minimum, this maximum. Now it groups all of the peaks together. So it actually looks across your data, and in a targeted setting where you don't have things like MS IDs from doing a DIA umpire style analysis, you don't have identifications yet. Skyline's going to go through, it's going to look for the top 20 peaks for each target in your window. The peak grouping that it's doing here, it's doing the exact same thing that we do by eye. So it's looking at the elution profile. So that's the time dimension. It's looking at the start retention time, the end retention time, and the top, the apex retention time. If all of these fragments belong together, then they should all have about the same retention time. They should all elute the same. Peaks with similar values get grouped together, and Skyline widens out those boundaries one more time. So now it's taking the boundaries for all of the peak group together. So now a little bit of building on things that Brian had just said. So now Skyline has top 20 peaks, but which one is actually the peptide? This gets into that peptide identification, the peak identification. So Skyline takes a lot of different scores. Same type of thing that we do with our eyes. We're going to be doing that in just a little bit, but it's going to take a lot of these things and for each peak group, tally up a score. The top scoring peak group is what it picks to be the peptide peak. So you might notice if you're familiar with DDA scoring and like mProfit algorithm, a lot of these are the exact same. So it's looking for things like the shape of the peak, the coelution. We just mentioned how the minimum, how the start, the end, and the apex retention time should all be the same. It's scoring that. It's scoring the intensity. More intense peaks are more likely to be the peptide, Skyline says. And then it tallies up all of these scores, and that's how it assigns the right peak group. So what does this actually look like? So in this processing, when we look in Skyline, we'll see chromatograms that look something like this. So in the Skyline software, on the horizontal axis, it's the retention time. That's the time the peptide's coming off of your column. On the vertical, it's the intensity. Intensity being kind of a little bit a proxy of abundance. And then each one of these colored lines is another fragment that was measured for that target. And each fragment is colored differently. And then the legend tells you the fragment identification, the name of the fragment. That's the Y8, Y6, Y4. And then it tells you the mass to charge of that fragment. So here I'm showing you two chromatograms in two different situations. The first situation on the left, this is a very easy call. It's very easy as a mass spectrometrist to look at the chromatogram on the left and say, this is definitely the peptide. So we're looking at things like the coelution of all the fragments are together. It makes this kind of rainbow shape. They're all stacked. The peak is very symmetrical. Compared to the chromatogram on the right, it's a little bit messier what exactly is going on. There's a second elution of this peak group, it looks like. That could be for a lot of different reasons. The chromatogram shape is a little shakier. It has a big tail on it. So when we look at these two, ideally, we want to see the chromatogram on the left as opposed to the chromatogram on the right. So Skyline's really good at detecting when a peak group is very clean, where it's very obvious. So here's one peptide from a yeast protein. If we look at this peptide across the full chromatogram, so across the entire experiment, there is really only one peak that looks like it's this peptide. It's a very strong signal. It's a very intense signal, and the fragments are all stacked very nicely. On the other hand, like Brian just showed us in real data, a lot of times a peptide doesn't look that good. So here's another chromatogram for a different peptide. You can see there's a lot of possible signals in here that could match to the peptide of interest. Only one of them, for simplicity's sake, only one of these peak groups is going to be the right peptide. So how do we know which one's right when it's such a mess? We can use things like libraries. So if we have a library MS2 Spectra, whether this is a predicted one using PROSIT, or whether this is a DDA MS2 Spectra library that we've acquired previously, or it's a repository spectral library that we downloaded from NIST or one of the sources that Brian gave us earlier, Now we can use something called the dot product. So the dot product is just saying, based on the library spectra, how well does your measured spectra correlate? Higher is better. The perfect number you could get is a dot product of 1. The lower the dot product gets, the less likely that the peak group is correct. It doesn't match your library spectra. So this peak group at 16.4 versus the peak group at 23.9, they're both pretty intense. 16.4 is actually more intense, and the chromatogram shape looks pretty good. But if we use the dot product, the peak group at 16.4 only matches the library spectra by like 82% match, versus on the right, the peak group at 23.9 minutes has a dot product like a match of like 99% match. So out of these two, the right peak group is the one on the right. So this is kind of what these softwares are doing when we're analyzing DIA data. It's taking all of these things together. It's taking, if you have a library spectra, it can use that library spectra, match it within your own data and say, oh, even though this data is really messy, this peak group over here looks a lot closer to the library. And this is why it's so important, if you have a library, that it matches as close as possible to your actual samples, because the fragmentation and interferences in your samples might be different, and the same instrument, because the fragmentation energy, like Brian mentioned, is a little bit specific to each instrument. Now, that doesn't always save you. Sometimes, even if you do this, it can be important to manually curate your data, because things like interference. So here's 2 examples of interferences in peptides where you can see Skyline picked the right peak. Over here, I picked the wrong peak, actually, on the right-hand side. But where the peak is, there's still a fragment ion that is contributing signal that's pretty noisy. So you want all of the signals to be pure signal, not noise. So even if you run these algorithms, even if you have Skyline analyze your DIA data, it can still be very important to manually look at it and convince yourself that things that are statistically significant are really truly biologically variants, and not, for example, statistically significant because there was one of these interferences in your data. So this is something that we'll be talking a lot more about later in the program as far as doing statistical analysis, but it always benefits you, even after you do a statistical analysis and you get the things that are statistically significant. It's always nice to go back into the data using something like Skyline and convince yourself, before you send the biologists off on a wild chase for a protein that's not actually differential, convince yourself that the chromatograms are real. So we talked a little about these criteria for picking signal. It's the same thing we do as the expert. It's the same thing that Skyline is doing behind the scenes in all of these softwares. I'm going to just review these real quick, and then I'm going to take a couple questions. So we talked about we're looking for the coelution of all of the fragments being measured. That's making the nice stacked rainbow. Talked about the peak shape. Symmetrical is best. We talked about signal intensity. The more intense the signal, the more likely it is to be the peak, but not always. That requires no prior knowledge, no previous work. If you have prior knowledge, it makes it so much easier to pick the right peak. So if you have an MS-MS library spectra, that gives you that dot product value. If you set up retention time standards, indexed retention time calculators, that can also speed up the process and be more sure that you're picking the right peak. Not super relevant for DIA, but if you have those stable isotope labeled standards that someone had mentioned in the chat, that helps you pick those paired endogenous peptides a lot easier, but it doesn't scale necessarily to most DIA applications. It works really great if you have targeted applications like a PRM having a heavy labeled standard. So I wanted to pause real quick before we go on and clear out some of the questions. So the MZ in Skyline, 99% of the time when you're looking at the mass to charge, it's going to be the fragment level. Skyline does include precursor level information. You can pull in MS1 scans and look at the isotope distribution for your precursors. 99% of the time in Skyline, we're going to be looking at fragments though. We got the no enzyme question. So if you're not using enzyme, digestion enzyme, sorry, for if you're doing something like immunopeptidomics or venomomics, you can add a no enzyme. You could also just skip that whole step and type your targets directly into the document. So just right on the left-hand side, you can add targets simply by typing on your keyboard, whatever peptide sequence you want to look for in a targeted setting. Should isolation scheme be the same as your settings on the Orbitrap? Yes, when you're analyzing the data in Skyline, and Brendan will be doing this more tomorrow too, with the tutorial. Your isolation scheme, you should tell Skyline how it's doing it. Skyline also has an option where it can read your scheme directly from your data files. So you don't necessarily have to have that setting pre-baked into Skyline to read in your data. You can have Skyline read the isolation scheme directly from your file. Can Skyline identify how many protein groups? No. So great question from Bowen. Skyline actually doesn't do any protein parsimony step like that. And it will give you options. In Skyline, you can have options where you can add a non-unique peptide, a duplicate peptide. You can add it to just one protein, or you can add it to all of the proteins. But if you want to get back at parsimony and protein groupings, you'll have to export Skyline and, for example, put that data into something like MSStats to regroup your proteins from a DIA raw file. DIA Skyline can detect peptides and all of the proteins that it came from a DIA raw file. So you're going to be doing that tomorrow with Brendan. Yeah, I think Juan's going to get this question from Hugo on acceptable value for a dot product. The short answer, the short version, it depends. Higher is better, but there's a lot of reasons why you might get a low dot product, a poor dot product, but it's still the right match. So I think Juan's got the rest of those. But so that's kind of like the characteristics that if we were to analyze, for example, PRM by ourselves, by eye, it's kind of doing the exact same things that these softwares are doing, and the software is just automating a lot of this. So prior information. So we can add prior information to our Skyline document using the settings, peptide settings, and there's this library tab. And you can have as many libraries in here as you want. If you want to activate that spectral library, kind of turn it on, you just check the box. We're going to be doing this in a bit using kind of like a fake repository spectral library. Once you have a library in here, you can hit this explore button and it'll show you the spectra for all of the peptides. So a lot of times the quality of the spectral library will make a big difference in your data analysis. So some peptides will fragment very well. So that's this bottom right-hand side. This is an example of a really nice fragmentation spectra. What makes it nice compared to the one on the left? There's a lot of fragment ions that span kind of the full MZ range. So if we look at the one on the left at the top, there's only a couple identified fragment ions in this spectra versus the one on the bottom right has many fragment ions that span kind of the full length of this peptide sequence. So this is another thing that you can do before you even go to analyze your data. If you're doing a targeted analysis of your DIA results, you might look at the peptides that you're interested in your targeted analysis, and they go in the target pane on the left-hand side, as I said earlier. And then you can go through and check the spectral library before you pull in your DIA data. And if it's a garbage looking peptide like the top left, maybe you remove that from your analysis. You don't even bother trying to reanalyze this because it's probably going to look like garbage. So you can kind of cut down before you even look at the DIA data based on your prior information that you have. Okay, so that's library spectra. We talked a little bit about that. You can see some of the papers for how to build different types of library spectra. But let's talk a little bit more about this correlation to retention time, because we got a lot of questions about that in the last section. So retention time, why should we care? Because peak picking is a lot easier when we have retention time information. So in Skyline, this is kind of an example of a target. So you could just type this peptide sequence into Skyline, and it will tell you the precursor mass to charge value, and it'll tell you a bunch of the fragment mass to charge values. Predicting MZ values is extremely easy. Predicting retention time is not so easy. So having prior knowledge of when a peptide elutes helps your software pick the right peptide peak. This is the same if you're using Skyline or if you're using, for example, Spectronaut in those Biognosys IRTs. That's why they like when you have them. The more information the program has, or the operator has, the expert has, the more likely you're going to pick the right one. So one kind of like thing I want you to recall from this, if you take nothing else away from this section, is that empirically sourced retention time is better than predicted retention time. So we're going to go into why that is, but if you take nothing else home, the take home message is that empirical sourced retention time is better than predicted. So we can look at retention times in Skyline just by selecting all the targets. It'll show you the full retention time chromatogram and show you where all of the targets are. Just kind of a nice little trick. But the way that we can get retention time information, Skyline has two options built in. The first option is called SSR Calc. This approach, SSR Calc, is sequence-specific retention calculator. SSR Calc requires no prior information. It'll just take the peptide sequence and it will add all of the retention coefficients for each individual amino acid, and then add some special consideration for whatever amino acid is the N terminal. So SSR Calc does pretty well. In Skyline, this is right built in. You can see the SSR Calc visualizations in the retention time window. In the retention time window, if you go to graph, regression, score to run, it'll show you this SSR Calc score. It looks something like this. So now on the horizontal, we have the SSR Calc score from 0 to 50 here. And then we have the measured retention time. So this is the actual retention time for these peptides on our LCMS system. So a couple things. We've got the SSR Calc score. We've got this real retention time. So now each one of these points is a peptide. Notice the SSR Calc score is not supposed to be an exact value of the measured retention time. It's just a score. And the correlation of that score and the correlation of the retention time is really all it's doing. So for example, this first peptide that has a very low SSR Calc score also has a fairly early measured time. The higher the SSR Calc score, the more likely that peptide is to elute later, have a higher retention time. So that's really all the SSR Calc score can tell you. It can't tell you a very exact, precise retention time. It just kind of gives you an idea. We can also compare how well the SSR Calc score works for our data, looking at some of the metrics up in the top here, for example, the R value. But really all this can tell us is if we click on a peptide in the SSR Calc score window, we click on that peptide, it'll take us to that chromatogram. This one correlates very well to the predicted retention time, but maybe we want to click on some of these peptides who had an SSR Calc score that didn't correlate with what we actually measured. That might be an indication that the peak is not picked correctly. So that's really all we can do with the SSR Calc score. We can kind of look at the trend between the SSR Calc score and the measured retention time and try to zoom in our attention on some of these peptides that did not correlate very well. So this is a very simple example, right? But as we add more targets, this gets messier and messier. So now we get a whole bunch of outliers, and we're not even really at like DIA scales yet. This is just like a somewhat large PRM experiment. But you can see already there's some extreme outliers here. There's some things that didn't have any measured retention time. Like we should go back in our data and manually curate all of these to make sure they're picked correctly. Or we use empirical retention times. Rather than just using a predicted retention time, we can use the indexed retention time, IRT. So this is a really not an easy read paper, but I found it like a very intuitive paper, not a super tough read. The Escher paper from oh my goodness, almost 10 years ago now. It's a good one. IRT, as Brian mentioned, there's like kind of like the trademark IRT, and that's just a set of peptides to use for this technique. But IRT kind of itself is just an approach that stores a calibrated, empirically measured retention time in a library. Then we can use it for future retention time prediction. So this means for future, it means it doesn't have to be on the same LC system. It doesn't have to be the same chromatography gradient. And that's really why it's so powerful. So how does that work? So let's imagine we have 3 peptides. 2 of these peptides are going to be IRTs. This can be anything. It doesn't have to be a kit of synthetic IRTs. It can be anything in your sample. So you have 2 anchor peptides, IRT peptide 1 and IRT peptide 2. Somewhere in between falls your peptide you're interested in, peptide X. So what IRT does is it says, anchor 1, set that arbitrarily to a value of 0. Anchor 2, set that arbitrarily to a value of 100. So now we have 2 anchors, 0 and 100. Whenever peptide 1 elutes, that's value 0. Whenever peptide 2 elutes, that's 100. Peptide X elutes 60% of the way between these 2. Because it's 60%, we'll assign it IRT value 60. Now, if I change my gradient, the percentage, these index values, doesn't have to change. I can change my gradient, and even though IRT peptide 1 now elutes at 10 minutes instead of 5, and IRT peptide 2 elutes at 50 minutes instead of 25 minutes, if we set those still at 100%, we know that peptide X elutes 60% of the way in between on this linear gradient, and therefore has a new measured retention time value of 34 minutes. So that's really all IRT is doing. You can set these red anchor peptides to be anything you want, as long as you measure them in both runs in all of your mass spectrometry acquisitions. As long as those anchor peptides are there, you set up the calculator one time, and then after that, you can apply that calculator in any other samples. So Skyline can set this up. A lot of the other DIA software also kind of performs a similar thing. For example, Spectronaut, I mentioned those Biognosys peptides you can purchase and spike those in to use as IRTs. Other software will just use any peptides that are measured reproducibly in all of your samples and align the retention times using that. It's pretty clever. So in Skyline, you have this IRT calculator and you set this up. When you're doing your data analysis for DIA in Skyline, this is kind of done for you. It's kind of folded into a tutorial, but this is what it looks like. And this is just taking those IRT peptides. You put your standard peptides, that's the red ones, you plug those in. You set up the calculator, assigning them a value, and I can link a tutorial for you if you're interested in setting this up yourself. And then you measure all of your peptide X's, go down in this measured peptides window. And then it just, in your calibration run, it'll assign the IRT values to your standard peptides just arbitrarily, 0 to 100. And then for your measured peptides, your experimental peptides, it'll just plug those in based on that linear fit on your lines. So it's pretty powerful. Using IRT, you see these in Skyline the exact same way we saw the SSR calc, except now instead of having SSR calc score on the x-axis, on the horizontal axis, we have the IRT value. The IRT value, like I said, can span whatever you want. It's not supposed to be a direct one-to-one value with a measured retention time. It's just to get the correlation. You might notice right off the bat, if we compare that back to the SSR calc score, look, these peptides just fall a lot nicer onto this line. And that's the exact same thing that Escher found in her paper, where the SSR calc retention time, just based on sequence prediction, is not as tight as using this empirical index retention time. So that's kind of why a lot of the DIA approaches prefer if you use kind of like this IRT style or build in some way to include retention time as prior information in analyzing the data. Okay, so we're going to switch gears here in just a bit. So let me just double check any questions that we have open. So Susanna asks, does IRT work when your retention times do not change linearly? It does get a little trickier, but Skyline does have other fits besides linear fits. So you can definitely play around with that. I think most people do not get quite as fancy with their liquid chromatography. So first, I commend you for doing nonlinear gradients and maybe more interesting multi-step gradients there. But yeah, Skyline has nonlinear options for retention time calculators. Adding on to a question, what is the difference between remove duplicate peptides and remove repeated peptides? So if you remove duplicates, it'll leave a peptide in one protein, the first protein. If you remove repeated, it'll remove any peptide that exists more than once in the document from everything. So for a lot of targeted work, we like to remove those. For a lot of DIA work, you might not want to remove those because you want to do post-processing that's going to regroup your peptides into proteins. I allude to manually curating peptides. It seems reasonable in PRM when there's maybe 100 peptides, but wouldn't we let the software do this with DIA? Absolutely agree. That's exactly what I do, Leanne. I will process all of my data all the way through statistical testing, and then anything that's significant statistically, which tends to be closer to like the 100 peptide limit for manual curation. Anything that's statistically significant, I'll go back and double check those chromatograms just to make sure that it's real signal. Sometimes it doesn't look great and it's not something I would want to send a grad student or a postdoc chasing for their research project until I convinced myself. So absolutely agree. Manually curate afterwards. Sometimes what I'll do with DIA is I'll spot check a couple that I know should be interesting in my data, just to make sure that the software looks like it's working correctly. Can I use a Spectronaut library in Skyline? Skyline's library building takes a whole mess of different inputs. So I will find a link, or maybe one of the TAs can back me up and get you a link to the BLib BiblioSpec BLib builder list of software that it will take for spectral libraries. Question from Hugo, if I calculate the slope and the axis, intercept, yeah, on my system with IRT, can I use these values instead of IRT? You could, but I think you still need to kind of use those index values. I mean, that's kind of how IRT works, is it's taking that slope and it's taking the fit of these anchor peptides, refitting those anchor peptides, and then plugging in where a experimental peptide should fall on that line. So I think you're describing exactly how it works. I think so. If you had more questions, let's follow up again later. But yeah, I think you're just describing exactly how IRT works. So good intuition.
Lindsay, I just wanted to add one thing with the library. I just shared the link, but I actually don't think this list has been updated in a while because I don't see MS Fragger there. So there might be the possibility that there might be some tools that actually Skyline supports and has been discussed in the support forum, but has not been updated to this list. So give it a try or ask in the support board.
Yeah, totally agree. The support board, hopefully Juan can drop a link somewhere. The support board, search your questions there because odds are someone has definitely asked the same question before, and they're usually very quick. Brendan and his team are very quick to get answers back to the community. Okay, So I think I'm going to move on a little bit just because a couple of these questions I think we're actually going to get to when we do some of the hands-on. Okay. So that's IRT. Skyline will take any data from any vendor. So once you get these things set up, you get your spectral library set up, you get your IRT calculator set up, you can pull that data in from any vendor's software and get your data processed. So that takes us, now your data is in Skyline, you're going to do some core analyses. There's a couple built-in native visualizations in Skyline that make it very nice for curating your data. So we already talked about looking at chromatograms. Skyline is really kind of the bread and butter of Skyline, is being able to visualize these chromatograms and manipulate them. When we look at this in the Skyline software, it looks something like this, where on your left-hand side, you get all of the targets for your experiment. So you set these up prior to bringing in your data. You have to have the target set before you can import your data, or else Skyline doesn't have any chromatograms to extract. So you'll see that it's built at like a hierarchy. It's got this tree where it's the protein. Underneath the protein is all of the peptide sequence. Underneath each peptide is a precursor. So this is the MZ value for that peptide sequence with that charge. The 2 pluses here means it's a +2 charge precursor. And then underneath the precursor, you have all the fragments that you had set up in your settings, transition settings. When you click on a peptide or a precursor, it will show you the chromatogram for that target. And like I said before, the retention time on the horizontal, the intensity on the vertical, and then each one of the colors corresponds to a different fragment ion. I do have an aside here too. You can show the precursor ions as well instead of just the fragment ions. There's a whole tutorial for that that I could link if you're interested in doing that kind of thing. So we have our process data. We talked about these 3 things that we're looking for in our data, talked about how adding this prior knowledge is very important, especially for DIA to have library and to have retention time information. We talked about how interferences can be an important reason why we need to look at our data. We might need to remove those interferences in order to get good quantification values. Skyline's really good at picking easy peaks. However, DIA does not have easy data. So how can we be sure that what Skyline has picked, or what really any of our software has picked, is real? Using some of these visualizations helps us very quickly pick out, even if we have a lot of targets, when something might have gone wrong. So right now I'm showing you a retention time replicate graph. So this means the retention time for that peak group is shown on the vertical axis. The box plot represents the start, end, and apex for that peak that was picked. Each one of the columns here is another replicate, another data file that we acquired. So you can see for the majority of these data files, the retention time is about 31 minutes, give or take maybe 30 seconds, except for one. One of these replicates is an outlier. And this particular replicate, the peptide eluted way late. If we click on that box, it'll take us to that chromatogram. And we might want to double check, did that peak get picked correctly? Is that peptide really eluting several minutes later than the rest of this cohort of samples? If not, maybe we want to look at the 31-minute retention time window and see if there's a peak there that did not, for whatever reason, score high enough to be picked. Along the same lines, we can look at the peak area percentage. So this is showing, the stacked bar chart is showing you each fragment that we measured for this peptide, and it's showing you the percent breakdown of the peak group. So for this particular peptide, the largest, most intense fragment is the red Y6, and then the teal Y4, and then maybe kind of a three-way tie between the green, the orange, and the blue, with the purple Y9 making just the smallest percentage of the peak area, except for this one replicate. So this one replicate has a very different fragment distribution. There's almost no red Y6 in the peak group that was picked for the sample, combined with the fact that it was picked so much later in retention time from the others, that would indicate something that I would want to manually follow up on and make sure that this peak is picked correctly. This can be very important downstream when we're trying to do things like MS Stats, because MS Stats, we're going to be trying to compare the variance between two sample groups. If some of the variance in a sample group is due to a mispicked peak, it can make it impossible for us to detect that statistically significant biological variance. So this is just to show you that even if a software is doing a really good job picking peaks, going through and manually curating for some of the things you care the most about can be very important in getting the results that you hope to get. So that's just a couple of these manual curations, and I think we're doing that a lot more tomorrow too. But last, before we moved on to doing some Skyline ourselves, I wanted to point out there's a lot of additional analyses for doing things like biological inference and visualizations and statistics. So there's just a whole slew of different tools here that you can add on, kind of like apps on top of Skyline. To get these, you can add these tools just by going to this tools tab up here and going to the tools store. And if you go to add, you'll see add from tool store has a whole bunch of different options here that can be very helpful in your research. For example, I had seen one person ask a question about isoforms. This population variation tool in the middle there takes data from, I think it's dbSNP. So if you're in the clinical realm and you're looking at some particular protein and you want to make sure that you're measuring all of the peptides and single amino acid variant isoforms in the population, in the human genetics world are included, you can use that tool to very quickly do that. There's a whole bunch of other tools, including tools by Brian, including tools by Olga. So definitely something that's worth checking out so that you could do things inside of Skyline. Okay, so that's kind of reached the end of what we wanted to mention as a very quick wrap up for this kind of lecture-based section. Targeted assays or DIA assays, Skyline handles them both. Vendor neutral, no matter what instrument you're on, you can analyze your data. A whole slew of additional software that you can add on top of Skyline-like apps, building in tools like IRT calculators and spectral libraries. We didn't even really get a chance to talk about some of these other community-based tools, including Chorus and Panorama. Panorama for sharing your Skyline documents. But there's a whole bunch of resources on the Skyline website. So definitely check it out. Check out the support boards like Juan had said. And kind of the three main takeaways, hopefully, that you got from this is taking some of those core concepts that Brian had mentioned earlier, putting them in context in the Skyline software and what they mean in Skyline, thinking more about the quantitative LCMS data, and especially why it can be kind of challenging with DIA as far as interpreting chromatograms and processing that kind of data. And then lots and lots of literature in there. Hopefully this gave you a lot of jumping off points from your own work. And main takeaway message, let's now try to apply some of these things in Skyline. While I get that booted up, see, thanks to the TAs and Brian for getting to a whole bunch of the questions. I'm going to get Skyline booted up now. So I'm just going to open up Skyline. If you're using Skyline daily, you will probably see a whole bunch of these pop-up windows. Skyline Daily, just to clarify, is kind of like the beta version. It's like the latest, greatest, all the newest things that Skyline has incorporated into the software. New features, bug fixes get included in Skyline Daily. Some of you, it sounds like, had some issues with downloading Skyline Daily, and that's because typically you don't have administrator rights on your computer. So hopefully you found a stable release that you could do. I'm going to tell Skyline to remind me later. And for this tutorial, okay, so for this tutorial, we're just going to do kind of a pretend example. This is really to kind of get you familiar with the Skyline software, because tomorrow Brendan's going to like take you into the deep end and go straight to analyzing DIA data. So let's build a little bit of a level of comfortability of using Skyline. In this little mini tutorial, we're pretending we're doing a targeted analysis of these 6 different plasma proteins. So let's imagine, you know, your boss says, I want an assay for these 6 proteins, or let's say you have, you know, you want to go back and reanalyze some plasma DIA data. There's this, maybe an awesome paper just came out. They've got this plasma or this CSF DIA data set, and you're interested in these 6 proteins and you want to reanalyze their data for your 6 proteins. What are some of the first things that you would do to set up Skyline? So the first thing, the very, very first thing you can and should do is to go to Uniprot and get the FASTA. If you don't already have the FASTA for these, it's like kind of the basis of almost all the mass spec that the world does is get the FASTA. So you navigate to uniprot.org. If you want to play along at home, feel free to pull this up on your monitor. If you want to watch me do it and then do it again, the tutorial has screenshots and step-by-step in the PDF. So feel free to, if you want to watch one and then do one yourself later, go for it. If you're a hands-on learner, I'll try to narrate everything I'm doing. So up in the top left corner on the uniprot.org website, there's this retrieve/ID mapping. If we click on that, it takes us to this page where the very first thing it says is provide your identifiers. So here we just type in all of the proteins we're interested in. So I'll type in ApoA1, ApoA2, ApoB, ApoC3, ApoE, and KNG1. If you want, if you have your own pet protein that you'd like to do this on, feel free to type in your own pet proteins that you're interested in. Scroll down to the bottom where it says this 2 select options. What we have just typed in are gene names. So we'll scroll down in that drop-down menu and select gene names. We want to map this to the Uniprot KB and the organism. If we start typing in human and we wait a hot minute, it should pop up Homo sapiens. So again, screenshots walking you through this are available on the course website. We'll hit submit. So you may notice it mapped to 47 Uniprot KB IDs. On the left-hand side, we're going to filter by reviewed. So this is the Swissprot reviewed, and now we have our 1, 2, 3, 4, 5, 6. We'll hit download button, kind of in the kind of top leftish, middle leftish, I guess. Download button, make sure download all is selected. The format is going to be this FASTA, the canonical. You might notice if you open this up, you can do canonical and isoform. If you want to play around with that during this tutorial, feel free to do that. I'm just going to do the canonical. I'm going to download it as uncompressed because I'm lazy and I don't want to unzip it after I download it. Hit go, and you should see it downloaded. FASTA looks like this if you haven't seen a FASTA before. The first line of the sequence has the identifier for that protein, has like a short description of the name of the protein. Then it has details like the organism. And then the next lines after that are the protein sequence. So it has that with this little kind of like side caret delimiter for each individual protein in here. As you're scrolling down, you might already notice some of these proteins are quite large and some of them are not so large. ApoB is just quite large, many lines of amino acid sequence. Okay, so we have our FASTA. Remember where you saved it. You might want to rename it. I think a lot of proteomycists like to rename their FASTA files, for example, by including, you got it from Uniprot, Skyline tutorial. This was 6 entries and it was accessed on 2021-0503. So a lot of times, later down the road when you're looking to publish, a lot of times the FASTA, you want to have the details, how many entries were in your FASTA and when you accessed it. So saving those right in the file can be very handy in the future. Okay, got our FASTA, ready to go back into Skyline. We're going to open up a blank Skyline document. So we're just going to go blank document right here. Now you should open up, we had seen this window earlier too, but in this Skyline document, what we're going to do first is go to settings. Make sure you have the default settings checked. I'm going to double check that I do. We're going to go settings, peptide settings. This is just like how we had talked before. So peptide settings, you want this to match whatever your experimental system was. You notice there's a whole lot of options here, but you can also add. So someone had a question, can you add different types of enzymes together? This is where you could kind of program that in. Maximum miscleavages, you can set this to whatever you want. For now, for this tutorial, let's just say 0. We're not going to set a background proteome for this tutorial. You will be doing stuff like this tomorrow. Retention time calculator, we don't have any. For the filter, let's change this just for fun. The default settings are 8 amino acids to 25 amino acids for the peptide sequence. Just for fun, we'll change this to 7. And we'll change the exclude N-terminal amino acids to 0. This default setting is at 25 because a lot of times, if you notice when we were scrolling down through that FASTA file, all the proteins started with that initiator methionine. A lot of times all of that stuff kind of gets cleaved off or it's signaling peptides at that end of the protein. So excluding those N-terminal amino acids, a lot of times biologically they're excluded anyway. So we just exclude them out of the FASTA. Go to library. We don't have a library yet. We're going to do that in a bit. The default modification is carbamidamethylcysteine. We'll leave that in there. You may notice I have just a whole slew of other modifications in here for other Skyline work I do. For the maximum variable mods, we'll go ahead and set that as 1. You can also leave that in your document if you want. We're not going to touch the quantification. We'll hit OK. And now we'll go settings, transition settings. So notice this is a lot of settings up front. Settings, transition settings, we'll go to filter. For the precursor charges, we'll do 2 and 3, pretty standard. Because we have the +3, we'll add ion charges 1 and 2. For ion types, we'll add B's and Y's. And then for product ion selection, we'll leave from MZ precursor, but we'll change the 2 to last ion -1. And then we'll hit OK. So now that we have all these settings configured in Skyline, we'll go perform an in silico digest. So those settings are giving Skyline the parameters for performing an in silico digest. So if we go to file, import, FASTA, navigate to where you have your FASTA. I just saved mine right on the desktop. I named it UniProt Skyline Tutorial 6 entries. We hit open, and you should see the target list on your left populates with a whole bunch of peptides and proteins. If you look in the bottom right, I've got 6 proteins, I've got 326 peptides, 651 precursors, and thousands of transitions. So if I were to analyze this DIA data, I could try just pulling the DIA files directly in at this point. It would be a real pain in the butt if I were to do that because I have, as was mentioned in the Q&A, we've got hundreds of precursors, hundreds of peptides that we would be trying to manually curate out of our data with no spectral library, with no retention time information. This would be a real slog. I would not want to do this. So instead, what we'll do next, let's now pretend, let's open up a new Skyline document. Let's now pretend that we have some prior information. I'm personally not going to change my, save my changes, but you can if you want. Now we have blank Skyline document again. Now let's pretend we have a spectral library. We'll go to settings, peptide settings, library, go to edit list, and go to add. So we're going to pretend like we downloaded a spectral library, for example, from NIST. So we'll just call this May Institute SpecLib. This is just giving it a nickname in Skyline. And now the path, we'll go to browse. And we're going to browse to wherever you downloaded these data files, the tutorial files. So if we navigate to there, we should have, just finding where I saved mine because it wasn't on the desktop where I usually do. Okay, there we go. We have now moved these to the desktop. If we navigate to the Plasma Spectral Library folder, I already have a blib in here for you. So navigate to the tutorial files in the Plasma folder. Inside the Plasma folder, Plasma Spectral Library. If you open that up, you should have this NEU, Northeastern University Skyline Plasma Fraction blib. Hit open, and then hit OK in the Edit Library window. And then hit OK again. And then in the Peptide Settings Library tab, check the box for the spectral library that you just added. So now you should have this set. Box is checked, library set up, hit OK. And you should see kind of this library match window pops up. I'm going to move that just in the middle so it's a little bit bigger. Oh, you may have noticed I clicked and dragged that window. If you click on the tab and hold it, you'll kind of get this blue box that opens up. And then if you're still holding it, there's tabs on either side. So there's this kind of window over here. If I click and drag and hover over that, it'll snap it to the right. If I click and drag, I can snap it to the bottom. For now, I'm just going to click and drag and set it to the center. Kind of handy thing for when you're working with Brendan tomorrow to move the windows around to match what his Skyline looks like. So we don't have any targets in here now. We have this spectral library, but we don't have any targets. So we're going to do the same thing we did before. We're going to go File, Import, FASTA. We're going to go back to wherever that FASTA was. For me, I just put it on my desktop. UniProt, open. So notice again, now the left side target list window has populated, but it's a lot fewer things now. So if you look in the bottom right, we still have the 6 proteins, but instead of having 300 something peptides, we only have 141 peptides. If you click on a peptide and go, so if you click on a peptide, you open up the precursors by either clicking on this little plus sign, minus sign, or you can use your arrow keys. So you can use your arrow keys to tab up and down, and then use the right arrow to open it up, left arrow to close it. If you open it up, go down to a precursor, now you can see the spectra. So if you were doing a targeted analysis of DIA data, you might go down through some of these spectra and delete stuff that looks like junk. So far, all of these spectra look, not awful. So if we kept going down, tried to look for some spectra that looked really awful, maybe this guy, this particular peptide, AKP, it's not the best spectra I've seen. Maybe I would delete that so that I don't even bother pulling that data in because it looks kind of junky. So this kind of gives you an idea of how adding prior information gives us a better chance of analyzing our data quickly and correctly. So we went, if you had no prior information, just the FASTA file, and you tried to do this kind of analysis on just 6 proteins, you would have hundreds of peptides that you would have to curate and Skyline would be trying to pick from. If you add some prior information like this spectral library, now Skyline knows about half of those peptides that are theoretically possible are not empirically measured. So the more information you add, the better your DIA analysis will be. See, here's some more peptides that definitely have maybe worse looking spectra. So I would just hit the delete key and remove those without doing an analysis. So there are a couple questions. See, you guys are doing a great job grabbing questions for me. So talking about those duplicates. So we can go up here to the upper left hand, go to Refine, Advanced, and now you have options to remove repeated peptides or remove duplicate peptides. If you hover over that text, a lot of times Skyline will have help messages for you. So if you hover over remove repeated peptides, it will remove all the peptides except for the very first occurrence. So the very first protein in your target list will have that peptide and it'll remove all the other ones. Removing the duplicate peptides, if you hover over it, any peptides that are not unique will be removed. So out of curiosity, we can just hit that, see if there's any duplicate peptides in our mini list. There were no duplicate peptides.
Lindsay, before you move on, there was a question that is relevant to the window you had open about deleting peptides without a spectra, but yes, by math.
So in here, there's this final option too, where you can remove peptides that do not have a library match. We can click that. The way we set up this Skyline document, every peptide has a library match. If you set up your Skyline document differently, you could click this, hit OK, and it would remove any peptides that happen to be in that target list that did not have a library spectrum. But if you go if you go to settings, peptide settings, and go to the library tab that we were just on, here we have pick peptides matching library. That means Skyline's only going to add peptides to this document that have a library spectrum. That said, if we did something like pick peptides matching filter, you see that the number of peptides increased by like 50. So now we have a ton more peptides and not all of them have a library spectra. So you can tell which peptides have a library spectra or not on the left-hand side in the target pane. If you look real close, and this might be real tiny on your screen, I wonder if I can, it's already as big as it gets. So on the left-hand side, if you look real close to where there's a P and an oval and then kind of almost looks like an antibody or something, like a little red V underneath it. If you look immediately to the next side of that, some of the peptides have a little black and white spiky boy next to it. The spiky boy indicates that that peptide does have a library spectra. And you see these first two peptides for me on my document do not have the little black spiky lines. That means they do not have a library spectra. So if I open that up and I click on these precursors, I get nothing. Versus when I go down to these precursors, I see the library spectra. Up here, nothing. Down here, no. So now if we did refine, advanced, remove peptides missing library match, those disappear and get refined out of our document. Svetlana has a question. Why did we skip miscleavages? Just for fun, we can add those back in. We can go to settings, peptide settings, digestion, max miscleavages 2. So we see nothing happened in this document. I think what we have to do is settings, peptide settings. Change back. So remember before we did settings, peptide settings, and set it to filter. If we set it back to library, there we go. So you can add as many miscleavages as you want. The more miscleavages that you add, especially when you're talking about stuff like DIA, you just increase the search space, and it can get very slow to process that data. Aaron has a question. In Skyline, once you have your target list populated and all your sample results uploaded, is it possible to filter out peptides within a protein by correlation with each other? So there are some advanced refinement things up here in Refine Advanced that you can filter out based on things like, oh, I don't have it in here because I don't have data in here right now. I don't have data in this one, but I might have another document that I can just open up to show you real quick what that might look like in another example. So here's a slightly more advanced Skyline document, but that's okay because you're going to be doing all kinds of crazy stuff soon, right? Okay, so slightly more advanced Skyline document, but it's something that I had on hand. This Skyline document, if we go refine advanced results, we have a lot more options in here to refine our results. So once you get actual data in your Skyline document, a couple more options like kind of unlock for you. So there's not really anywhere in here to do a correlation, an abundance correlation. So you would have to export this, do it in Excel, and then refine your peptide list here. But there are some other things in here that are maybe kind of nice, like refining by the CV or refining by retention time correlation. There is nothing for abundance correlation, however. So that one you would have to do outside in Excel, pull your data back in. It's not awful to do that. If you do that, you can file export, or there's an option in here, view document grid, and then you can change your report setting to peptides, for example, and it'll have the abundance measure for different peptides. You can hit this export button in the document grid to export, and then edit, or sorry, edit, insert peptides, and you can just insert whatever peptides you want to keep in there. It's definitely a workaround. So there's nothing directly for abundance. Andrew Reed, how is abundance measured in Skyline? Abundance is the MS2 chromatogram. So let's see if I can find a good one in here. Okay, so here's maybe a decent example. Let me make this bigger for you. Not a decent example. Here we go. this guy looks good, but has a giant interference. So Skyline will take the sum of the MS2 fragments and subtract the background area. So all of these chromatograms that you see in Skyline always start at 0. That's not because it's truly from your instrument starting at 0. Skyline's performing background subtraction and then showing you the background subtracted peak areas. When you're calculating these peak areas, Skyline's also including any interferences that are in here. So this big purple guy looks awful. Probably we should try to find where that purple guy is. It's a Y1, no surprise. And make sure he's not quantitative. So you can right click on the fragment and hit the quantitative tab. And then that fragment will no longer be used in calculating the peak area. So if I turn that on, you can see it adds a whole bunch of area under the curve to the peak area. Turn it off and now it's not being used for quantification. Skyline's also using these integration boundaries. So you may notice on either side, the integration boundary here was a little bit wider than the peak itself. So you might want to refine your data and pull that integration boundary in closer. So another kind of argument for looking at your data and trying to determine if your data is real or not before you run off and try to do statistical analyses. Is abundance normalized? Abundance in Skyline is not by default normalized. However, you can set normalizations. If you right click in the peak area window, you'll see this option normalize to, and you have a whole bunch of options. If you have heavy peptides, obviously normalize to your paired heavy standards, but you could also do normalizations like equalize the medians or do a total ion current. And I am positive you guys are going to be talking a ton more about data normalization later on in the course. So things to keep in mind. I think that just about covered the questions, but I will give it another couple minutes because I think I saw a whole bunch of questions kind of going through there. I think the TAs got most of the questions in there.
So we have roughly 5 minutes left, so please don't be shy.
Yeah, I intentionally wanted to leave some time at the end. But yeah, this was short Skyline introduction for your work tomorrow with Brendan. We'll be diving right into doing a lot more of this and especially analyzing the data. So kind of focusing on looking at the data tomorrow. More questions came in. So I think we got Janan's question on deleting repeated peptides and deleting duplicate peptides. In my data, it didn't do anything because we didn't have any repeated peptides or duplicate peptides. But yeah, the repeated versus duplicate is whether you delete all non-unique peptides or if you keep the first instance of a non-unique peptide. For DIA analysis on the whole proteome, should I use as target all the proteins in my library? Yeah. So when you're doing DIA analysis on a whole proteome in Skyline, you would use the entire FASTA file, and your library would ideally have all of the proteins in it. So instead of doing file import and having just this little baby FASTA file with just 6 proteins, you would do a FASTA file with the entire human proteome or a Arabidopsis proteome or whatever proteome you happen to be doing. And then when you make your spectral library or you make your DDA, your predicted library, whatever you're doing, use the exact same FASTA. Skyline does have ProCit capabilities now. So you can make a predicted spectral library directly inside of Skyline now. Use the exact same FASTA though. Can you see the MS injection time in Skyline? You cannot visualize the injection time, but I'll pull up this results file that I had on hand right here. You can see the points across the peak though, however. So down at the bottom here, next to the retention time, you see there's these red short vertical lines and then this little red number to the right, this little red 7. That's how many points were across this integration here. So if I made this integration really short, that number changes. If I make it a little wider, now I've got 7 points across the peak. Not great, not bad, but not great. IRT tutorial, let me grab a link for that. And I'll also post the IRT tutorial on the course page, the course Google Drive page. How does the program go from peptide to protein intensity? It just sums up. Peptides gets all the MS2 fragments get summed to a peptide. All of the peptide gets summed to protein. There are much fancier ways of doing this. For example, there's some options in Skyline group comparison that were really pulled from MS stats, like doing the Tukey medium polish. So I think you're going to be learning more about that later too. Definitely hold on to those questions, how summing to protein level might be different. Is there a way of automatically filtering out transitions based on peak width? I don't think in the refinement that there's a way to do peak width.
I think from the reports you can. You just have to find the correct report where you have a column with the peak width, and perhaps that I have never considered that actually. It will be interesting.
Yeah. So this is we got like 1 more minute. Let's see if I can real quick. So if you did that view document grid, you could potentially use some of these values in the document grid. There's average retention time, but there's a whole bunch of other values that you can play with in here. So Juan is kind of getting at the idea that you could probably use some of these metrics in the report to refine using this kind of data. It's not a single button though, unfortunately. Yes, Sean, the points across the peak are the number of MS reads across that peak. Yes, from the raw data, not the resampled data. Yeah, I think that's pretty much all the time we have, but if there were more questions, definitely put them in the Google document so we can try to get there. And the IRT tutorial will be linked into that Google document, so hit refresh. And hopefully that will get all of your questions, and hopefully that prepared you for the theoretical and the practical side of DIA for your sessions tomorrow.
Well, thank you so much, Lindsay, and thank you, Brian, as well. And thank you to all the teaching assistants who were dutifully answering questions in the background. So Juan and Diana, we really appreciate your contribution as well. Thank you very much. So tomorrow will be a hands-on session with Brendan. So make sure that you look over the documents linked to his session and prepare as far as the installations and downloading all the slides. And be ready, because Brendan has a lot of plans for you tomorrow. Thank you very much, everyone. It was really wonderful to see everyone today, and I will see you tomorrow.
See you tomorrow. Bye-bye.
Thank you. Bye-bye.